my lovely children out there. I hope all of you are doing great. Well, today I am here with a little gift for all those of you who are going to write your FA2 examination. And the gift is this video, because it will contain all the very important questions that could be probably showing up on the day of your examination as well. And I can guarantee you, your FA2 paper, biology paper, is going to go awesome. Yes, please do not judge this video by its length, okay? Judge it by its quality, and I am pretty sure it's going to be good. It is going to help you well. And my dear children, you just have to do one thing. Listen to me carefully. After 30 minutes, pause the video. You can write down your own notes and things like that. You can also get this particular PDF if at all you join our WhatsApp channel. So you need not worry about anything. All ears, all focus on me. That's all. Yes, are we ready to rock? Yes, I hope. And those of you who are having any self-doubt that I have not studied, I won't be able to do it, let me tell you, you are more capable than you think, and you will be absolutely able to do it. Starting with the first chapter that we have, which is Human Health and Diseases. I have taken 4 chapters. This, Microbes in Human Welfare, Biotechnology Principles and Processes, Biotechnology and its Application. If at all there are some other chapters also there in your FA2, you can put it down in the comment section below. Starting with the 1-marker questions, human immunodeficiency virus replicates in. Human immunodeficiency virus means you are talking about HIV and V. HIV virus, where does it replicate it inside our body? Where does it increase its number inside our body? Is it inside the B lymphocytes or NK cells? NK cells means natural killer cells. Or is it inside the helper T lymphocytes? Or is it inside the PMNL? PMNL stands for polymorphonuclear leukocytes. These are one such type of WBCs. You also call them as neutrophils. So where does HIV virus replicate? If at all you are given options like this, you need to understand that HIV virus are there for targeting T helper cells, because you know what, T helper cells are super important for maintaining our immunity, and HIV virus does not want our immunity to be strong, which is why it targets the T helper cells. It gets into the T helper cells, replicate itself, and then later kill the T helper cells, making our immunity go really low. So over here, your answer for this question would be C. But you need to understand one very important thing at the same time, that is, initially when HIV virus enters our body, it does not directly goes and affect the helper T cells. Initially one virus came in, now it has to increase its number. So the first site where it goes is macrophages. These are another type of WBCs. Another type of WBCs. So they go, white blood cells, okay? So they go into the macrophages, they increase their number, which is why I'll call the macrophages as what? HIV factory. It is the factory of HIV because they are replicating there. Now these HIV viruses will go to the main target, which is what? The helper T cells. Helper T cells are the target cells for HIV virus. Cool. So if at all you are asked what is the HIV factory and you are given an option like macrophage, please go with this. If at all this option is not there altogether, only helper T cells is there, please go with that. Okay, now if you have both these options and you are being asked that which is the target cell for HIV, then it will be helper T lymphocytes. These are the target cells. Now what is the diagnostic test that you use to detect HIV, my dear children? It is known as ELISA. In your chapter Human Health and Diseases, only two diseases have diagnostic tool. One is HIV. HIV's diagnostic test is ELISA, enzyme-linked immunosorbent assay. Okay, and then there is typhoid. For typhoid, the test is Vidal. For typhoid, the test name is Vidal. Cool. Now, do we have any treatment for HIV, ma'am? Is there a permanent cure? The answer is no. There is no permanent cure, but you can make sure that the person who has HIV lives for long with the help of some antiretroviral drugs. As in, HIV virus which is there, you call this as a retrovirus. You call this as a retrovirus. Now something against it will be known as what? Antiretroviral. So which is why we take the use of antiretroviral drugs for increasing the, what do you call, time span or lifespan of a person who is having HIV. Okay, so I hope this part of the story is crystal clear. You will be able to solve other questions also if any of this thing comes, right? Yes. Going ahead with the next question, the drug which is depressant and slows down body function is. You are being asked about a drug which is a depressant. Okay, it makes you depressed and also it slows down your body. Which is that drug? It is smack, is it crack, is it hashish, or is it charas? So hashish and charas, my dear children, they both come under some group of drugs which are known as cannabinoids. Okay, they are known as cannabinoids. Crack is the other name for coke, or you also call this as cocaine, which is again a different form of drugs. Now smack, which is there, it is the other name for one very important, what do you call, drug, which is heroin. So heroin's other name is smack. Okay, coke or crack is the other name for cocaine. Hashish and charas are coming under cannabinoids, which is a whole different category of drugs. So over here you need to know one very important thing. Where do you get smack from? Where do you get heroin from? So this is a plant. Its name is Papaver somniferum. Papaver somniferum. Now if you see, the things that are being released over here, you call this as the latex. So from the latex of Papaver somniferum, or you also call this as opium poppy, you get your first drug, which is morphine. Now this morphine undergoes a process which is known as acetylation. Okay, when it is made to undergo a process which is known as acetylation, it gives rise to the next drug out there, which is known as heroin, or you also call this as diacetylmorphine. You have a term over here, diacetylmorphine, because it has underwent acetylation. Now you have got heroin. The same heroin or diacetylmorphine is also known as smack, and it is a depressant. What about morphine? You told it's a drug, ma'am. It also has the same function. Is it also a depressant? Does it also slow down the body function? The answer is no. It is actually a sedative. That means you'll feel sleepy, and also it acts like a painkiller. Now, if at all they are asking you, doctors after surgery, they give some drugs to the patients after surgery who have immense pain. What is that drug? It is morphine, because morphine is a painkiller and also a sedative, so that the patients can sleep peacefully. But we cannot take it. It has to be a doctor who tells that, hey, this particular person has to be given this particular drug. Okay? Works? So this comes under a category of drugs which are known as opioids, because you get it from opium poppy, or what you call this as Papaver somniferum. So here I have made a list of all the drugs. You can go through it. So opioids, I'll just quickly go. Opioids, you have morphine and heroin. As I told, morphine is what? A sedative and a painkiller. Okay, and it is useful to patients who have underwent surgery. The structure of morphine looks like this. So I always say whenever there is a structure like O is for opioids, right? So you are talking about some opioid, and the name of this opioid is what? Morphine. Now this morphine undergoes acetylation. You got heroin, which is a depressant, and it slows down the body function. Now how do you take heroin? You can take by snorting or by injection. Snorting as least, or injection also. Okay, now how it is actually, it is white, it is odorless, it is bitter, and it is crystalline compound. You will also call this as smack. As I told you, you get it from the latex of opium poppy, which is also known as Papaver somniferum. Now one very important question comes, that is, where does it show its effect? On which part of our body? It affects our central nervous system, which includes the brain and the spinal cord, and it also affects the gastrointestinal tract. Okay, the gastrointestinal tract. So opioids are responsible for affecting CNS and also the gastrointestinal tract, and these are the examples. Done. Cool. And this is the picture given of Papaver somniferum in your NCERT. So a pink flower is there, and you have some pots out there. Okay, that's all. Now you have the next set of drugs, which are known as cannabinoids. Cannabinoids, where do you get it from? You get it from cannabis sativa. Now cannabis sativa, the leaf of it looks like this. And you know what? In cannabis sativa, every part of the plant can be used to form a drug. Every plant. So you say inflorescence of cannabinoids can be used for making drugs. Drugs like what? Drugs like marijuana, drugs like hashish, charas, ganja, everything, my dear children. You get it from flower tops, you get it from leaves, raisins, everything. Okay, so marijuana, hashish, charas and ganja. Over here, how do you intake it? There it was snorting and injection. Here it is inhalation or oral ingestion. That means you have it through your mouth, or basically you inhale it. That is the way you take this particular drug. Now ma'am, which part of our body does it affect? It affects the cardiovascular system. That means your blood circulation, everything is being affected with the help of what? This particular drug, which is a cannabinoid. And the structure of cannabinoid is also given in your NCERT. So as I said, for opioids, the O-shaped structure is given of morphine, right? Similarly, for cannabinoid also, the structure is given. If at all you focus, okay, there is like step-like thing over here. So you can remember, if at all this is there, then it is a cannabinoid. And one very important thing, cannabinoids have some receptors in our brain. This cannabinoid drug which are being taken, they go bind to the receptors, and now these receptors will be sending signal to our cardiovascular system. And if at all you are being asked this question that ma'am, which sort of drugs are being abused by sports person? So people who are in sports, they intake a lot of drugs, not all of them, yes, some of them, yes, and it is banned, by the way. But anyway, so you call this as abuse, okay, you call this as drug abuse. Now, which are the drugs which is usually being taken? It is cannabinoids. So cannabinoids are abused by sports person. Please remember that as well. Done? Cool. So cannabis sativa, the leaf, the cardiovascular system, and now we go to the next group of drugs, which are coca alkaloids. Coca alkaloids, my dear children, under that comes your cocaine. Cocaine is also known as what? Coke or crack. It is known as coke or crack. The mode of intake is again snorting itself. And now, where do you get it from? You get it from erythroxylum coca. You can get a question, which is a drug which you extract from a plant which is native to South America. So which is the plant which is native to South America? It is erythroxylum coca. So the drug's name will be what? Cocaine. Now, where does it go and show the effect? Again, the central nervous system, just like opioids. But here, in the central nervous system, they are specific. They are going to interfere between dopamine transmission. You know, dopamine is something that is released from one neuron. It goes to the other neuron and transmits the signal. It's a neurotransmitter. What it does is that it usually brings happiness. It usually brings happiness. Now suppose the receptors are being blocked such that the dopamine, its secretion is going on. So what will happen? Eventually we will get a sense of euphoria. There will be too much happiness. So it basically leads to what? A sense of euphoria. It gives us high energy, and if at all it is taken in more amount, it will also lead to hallucination. That means you are going to start seeing things which is not exactly happening. The dresses might be flying up, the ant might be looking like an elephant, and things like that. So those are hallucination that can happen. I know it sounds interesting, but it is not as interesting as it sounds. So coca alkaloids are basically cocaine, coke or crack. You get it from the plant native to South America, and that is erythroxylum coca. And it affects your central nervous system, especially it interferes between the dopamine transmission and stimulus. It gives a stimulus of high euphoria, but then later on, if it is taken in higher doses, it can also lead to hallucination. Now there is one more question that comes, what are the other drugs or the other plants that can lead to hallucination? So one is this erythroxylum coca. The two other plants which can lead to hallucination is Atropa belladonna and also datura. Atropa belladonna and datura are also something that has hallucinogenic properties. This is the plant of datura. Here you can see cup-like thing. Okay, datura having a cup-like flower out there. So funnel-like flower actually. So Atropa belladonna, datura, and also your erythroxylum coca are the ones which gives hallucinogenic properties. Cool, I hope you'll not forget that. So opioids, cannabinoids, coca alkaloids, and now there are some extra drugs given in your NCERT. But before I go into that, my dear children, if at all you are someone who is struggling with your PU2 preparation, we have launched an online crash course for you guys. And this online crash course is just for ₹999, a very feasible amount, right? So over here, what are the things that you will get? You will get one-shot lectures, there will be complete PYQ lectures, there will be subjects such as PCMB, apart from that Kannada as well as CS also. You will get your PDFs for all the lectures, there will be live and recorded classes, your doubt solving sessions are going to be ultimately live. There will be formula sheets given to you, and there will be a dedicated WhatsApp channel for keeping you updated regarding everything. The best part of the story is you are going to get a PU2 test series. Okay, question paper with answers and everything out there. So need not worry, just join the crash course right away. It's ongoing. It is going to be launched on, actually today. The first class will be starting from tomorrow. So do it quick. The link will be given in the description box. Yes? Okay. So as I was saying, there are certain other groups of drugs as well. Okay. One is barbiturates, amphetamines and benzodiazepines. Barbiturates, amphetamines and benzodiazepines. So BAB, you can remember that. What do? They are used for patients who are dealing with insomnia. That means they are not able to sleep. Okay, the people who are dealing with insomnia and depression, they are being given these drugs which are barbiturates, amphetamines and benzodiazepines. Listen, they do not cause depression and insomnia. They are used to treat. They are used to treat depression as well as insomnia. And this is often abused as well. People use it for no reason. Now there is one more drug given in your NCERT which you might not know that it is actually a very big drug. So whenever someone smokes, okay, basically there is tobacco. And the tobacco, what do you have? In tobacco, my dear children, it has nicotine. And this nicotine which is there, is a very toxic drug because it is going to affect your adrenal glands. The glands which are above your kidney. Okay, the glands which are above your kidney. And over here, the adrenal glands are responsible for production of two very important hormones, adrenaline and noradrenaline. Adrenaline and noradrenaline. Both of them are responsible for increasing your heartbeat and increasing your blood pressure. So in a way, when nicotine is being taken up, what happens? Your blood pressure as well as your heart rate increases. And it can also lead to cancers, respiratory diseases, gastric ulcers, lot of things. And you know what? When you smoke, there is a gas whose amount becomes even more increased, and that gas's name is carbon monoxide. And you know what it does? Usually our hemoglobin is bonded to oxygen, right? When CO comes, it makes the HbO2 break up. Now CO will be combining with Hb. Now so that means the hemoglobin is not carrying oxygen anymore, which is why we also feel that there is less oxygen. There is deficiency of oxygen in our body. So if at all a question comes that which is that drug which is leading to blood pressure increase or heartbeat increase, do not think twice. The answer for this question would be nicotine. It will be what? Nicotine. So ma'am, you said cannabinoids are the one that affects your cardiovascular system. That is there. But if at all specifically you are asked about blood pressure and things, nicotine is your answer. Okay, with that all the drugs are being completed. If at all you get any questions related to drugs, I hope you will be able to solve it. Going with question number 3, antivenom given against snake poison contains what? Antivenom. So whenever someone goes with a snake bite, doctor does not wait for their own body to, a doctor does not wait for the patient's body to produce its own antibodies, because it might take so much time to produce antibodies against the snake venom. So what do? When you go to the doctor, doctor gives you ready-made antibodies, or you call this as preformed antibodies. Why? Because you want your body to fight fast. That's why. So antivenom which is given against the snake poison contains antibodies, the ready-made antibodies. And if at all you are asked which type of immunity is this, okay, passive or active? It will be passive immunization. Always remember, in active you wait for your body to react. For example, I am going to go to a doctor to take vaccination against COVID. Okay, so in this particular case, I am being given some weak pathogen or some dead part of this particular pathogen, put it inside my body. Why? Because I want my body to release some antibodies. I want my body to release some antibodies against it. Anyway, this is not an actual infection. I am just making the scenario up, and it is dead and weakened pathogens that I am using. Why am I actually making my body produce antibodies? Such that when actually a COVID virus comes into my body knows what are the antibodies that it should produce, and it will help me out. That is the thing. That is where your body is included in vaccination, and that is active form of immunity. Over here, you are being given ready-made antibodies. Your body is not producing any antibody at this point of time, which is why you will call this as passive immunization. Always remember, this is an example of passive immunization where you provide ready-made antibodies to your body. Same thing goes with tetanus injection. So when we fall down from some place or things like that, we are given tetanus injection. Why? Because we want ready-made antibodies. So this tetanus basically is caused by bacteria. Now this bacteria can affect our body. We want ready-made antibodies to be there, otherwise they can cause a huge lot of trouble in our body. Okay, so antitoxins against tetanus also contains ready-made antibodies. Please remember that. Vaccination is an example of active immunity. So active and passive immunity, difference is given. Please go through it. Active immunity developed when the body produces its own antibodies. Passive means ready-made antibodies are given to the body. Now triggered by exposure to antigens. Active immunity comes from our own body whenever it is exposed to some antigens. Now this antigens can be through some natural infection or can be given through some vaccination as well. Whereas there, my dear children, we are being given, it's ready-made. Now over here, slow responses but long-lasting protection. Slow response is there because you're depending on your body, but if at all you see these antibodies which are released, they are going to provide me long-lasting protection because my body knows this has happened. Next time there are some memory cells also being formed, it knows that I have to produce the same set of antibodies again. But over here, when you are given ready-made antibodies, what is the problem? That is, they are going to end up someday. Okay, so they are going to provide fast protection, immediate protection, but for a very short duration. So please know the differences between both of them. You have some examples for passive immunity. Whenever you are given antibodies, okay, ready-made antibodies, this is an example of passive immunity. For example, you have how many antibodies? Gamed. 5 antibodies are there. The first antibody which is passed from the mother to the child is IgG through the placenta. The first antibody which passes from the mother to the child is IgG. This is example of passive immunity. Why? Because the baby's body is not producing antibodies. You are giving because it needs protection. Similarly, when the baby is being born from Amma, you give antibody A to the baby through the colostrum, which is the first milk. Okay, so first goes G through the placenta, then goes A from the mother's milk, that is colostrum. Okay, now suppose you are given some antibody injections such as in snake antivenom, things like that, that also becomes an example of passive immunization. Works? Now, since we are talking about antibodies, IgE is released during allergy. IgE during allergy. Please remember that as well. Fine. So, 3 antibodies function we should know. M and D, it's not been mentioned in your NCERT, so it's all cool. Now, fourth question, the substance produced by a cell in viral infection that can protect other cells from further infection is. So, there's a substance produced by a cell in viral infection. Suppose this cell has so many virus. Now, this cell is producing some substance which is protecting the other cells, which is basically protecting the other cells from virus attack. What do you call the secretion as? These secretions are known as interferons. Okay, you call the secretion as what? Interferons. These interferons tell the other cell that you have to be careful. It basically makes up our immunity a bit more stronger and makes sure that none of the other cells are being affected. So over here, your answer for this question is interferon. Please do not get confused with alpha interferon. Alpha interferon is given to patients who are having cancer. Why? Because alpha interferons are biological response modifiers. Biological response modifiers, as in they are also responsible for increasing our immunity, but this is given in the case of cancer patients. Always remember. So this is different, this is different. Okay. Now, which of the following plants does not possess hallucinogenic properties? Which of the following plants does not possess hallucinogenic properties? Erythrocysum coca, Atropa belladonna, Datura, Papaver somniferum. You are asked which is not possessing hallucinogenic properties. So we know Erythrocysum coca does, Atropa belladonna does, and Datura also does. What about Papaver somniferum? It is where you get opium poppy from, and it is nowhere responsible for what? It is nowhere responsible for hallucinogenic properties. Just give me a second. Yes. Cool? Next. Choose the correct statements. So it is about innate and acquired immunity. Innate immunity is accomplished by providing different types of barriers. First of all, let me tell you what is innate and acquired. So innate immunity means something that we have from our birth. For example, your skin, for example, your tears, everything. It is there with us from our birth. You do not need any pathogen to infect us, then only this particular things would be coming up. Is it like that? No. It is there with us from our birth, and it is non-specific. As in, basically it cannot say that I will let you in, but I will not let that particular organism in. So it is non-specific. It does not have any specific goal that I have to kill them, only them and things like that. No, it is non-specific. Now when you talk about acquired immunity, it is acquired through our lifetime. We get this immunity through our lifetime. Okay, so there are 2 cells involved in acquired immunity. So those cells are B and T lymphocytes. B lymphocytes and T lymphocytes. So ma'am, these cells are not there with us from the birth. They are, but they start showing their action only when we are getting infected. Only when we have an infection, that's when they start showing their action. And what is their action? B lymphocytes which are there, they are going to produce some antibodies. These antibodies are going to travel through my blood and capture all the antigens which are there. Okay, so which is why I'll say that B lymphocytes offer me humoral immunity. Humoral immunity. What is humoral? Something which is mixed in my humor. Humor is what? Fluid. So blood is my fluid, and in my blood I am getting this, through my blood I am getting this immunity, because in the blood is having what? Antibodies. So B lymphocytes give me humoral immunity. Talking about T lymphocytes, they directly goes and affect the cells which are being affected. Okay, for example, some virus attack is happening, they will directly go and kill them. But both of them, whenever they come across something, they are going to produce some memory cells. They are going to produce some memory cells which have the memory that this type of attack has happened. The next time it happens, I have to do this fast. Okay, which is why every time when we have an infection, the first time our immune system is very low. Okay, because it is for the first time it is meeting them. But the next time the same type of infection comes to our body will be fighting against it, because we have a memory of it. So T lymphocytes, my dear children, provide cell-mediated immunity. As in, here cells are involved for providing us immunity. These cells go tell other cells, and these other cells probably might be attacking, which is why you call this cell-mediated immunity. So under acquired immunity comes B and T cells. B cells produce antibodies, and these antibodies are going to mix with humor or the blood, which is why you say it provides humoral immunity. Whereas the other one is T lymphocytes, they give us cell-mediated immunity. Both of them, my dear children, we acquire it throughout our lifetime when any infection comes up. Okay, when any infection or vaccination happens, that's when all of them becomes active. Innate is something which is there with us from the birth. Acquired is something that is acquired through our lifetime, and acquired is specific. I know that this antibody has to be released when this antigen comes. So this is specific. Innate is non-specific. In innate, you have different type of barriers. For example, physical barriers such as skin and your mucus. They are physical barriers. Physiological barriers such as some secretions, your HCl in your body, or the tears in saliva which is there. All of them, they come under physiological barriers. Now there are some cellular barriers. Some cells are with us from our birth and they are very active. For example, you have neutrophils, PMNL. Okay, we studied initially, right? PMNL, which is neutrophils, natural killer cells, they are this, and macrophages, they are also like this. Okay, so they are the cellular barrier. Now cytokine barriers are there. There are some secretions which is being released from our birth itself, from the cells which are affected. Such secretions, example is interferon. Okay, so those are the few things that you need to remember. So innate immunity is accomplished by providing different type of barriers. Yes, there are 4 barriers, so this particular statement is correct. Acquired immunity is present from birth and is inherited from the parents. Is it present from birth and do we inherit it from our parents? No, it is not present from birth. Acquired immunity can be divided into antibody mediated and cell mediated. So antibody mediated, as in you're talking about humoral immunity only, because in humor you have your antibodies mixed. Okay, innate immunity is also known as specific immunity. I told you innate is non-specific type of immunity, which is why this is wrong. Acquired immunity consists of specialized cells such as B and T cells and antibodies that circulate in the blood. Is it true? Yes, ma'am. So I see 1, 3 and 5 are true, which is why your answer would be C. Okay, you might be asked questions to differentiate between innate and acquired immunity. Please go through it. I think we will be solving that question here as well. Cool? Chalo. Next, hemozoin is a. What is hemozoin, my dear children? What exactly is hemozoin? It's a toxic substance released from the plasmodium infected cells. So in malaria, you have this thing being released from the cells. So what exactly happens? I'll tell you, listen to me carefully. Okay, so there is a mosquito. Suppose there is a mosquito over here. In the mosquito's salivary gland, you have the plasmodium which causes malaria. You have the plasmodium which causes malaria, and this plasmodium is in a stage which you call as sporozoite. Sporozoite stage. So it enters our body in this stage which is sporozoite stage. Why do you think that the plasmodium which is there in the saliva, how can it get inside our body? So basically when a mosquito comes to drink our blood can become clotted very easily. and it does not want that to happen. It wants to drink it peacefully, which is why it releases its saliva. Its saliva has some anti-clotting enzymes which will not form clots. Now while it releases saliva, it also gives us what? The sporozoite stage of Plasmodium. Now the sporozoite is in our body. First it goes and affects our liver cells. First, it goes and affects our liver cells. In the liver cells, my dear children, they will undergo asexual reproduction. They will undergo asexual reproduction, and one time will be coming where the liver cell will be blasting open and all the cells are going to be released, and it is going to affect the next thing, which is our RBC. Now once they are inside the RBC, my dear children, what will happen? Again, they are going to undergo asexual reproduction. Asexual reproduction. Sporozoite came in, they underwent asexual reproduction. Now there is another stage, okay? You do not call them sporozoites again. Now this Plasmodium gets into RBC. Again they undergo asexual reproduction. And what stage do they give now? The stage which is being formed now is known as gametocyte stage. So ma'am, same thing will happen. It will burst open. Yes, it will burst open. And now what will happen? The gametocytes are going to be released in the blood. Now again one mosquito will be coming in. This mosquito will be coming in and taking in the gametocytes. When it drinks the blood, automatically the gametocytes are going to enter the mosquito's body. The different thing about gametocyte is that until now all of them were undergoing asexual reproduction, right? Now when it reaches this stage, that is gametocyte, the gametocytes are the one which will form the gametes. So ma'am, you are saying that gametes are going to be formed in our body? No. Gametocytes are formed, but they do not form gametes in our body once they get inside the mosquito's gut. When a mosquito drinks it, all the gametocytes reaches the mosquito guts. That's where it will be forming the gametes. That's where it will undergo sexual reproduction. So in our body, in the human body, no sexual reproduction. No sexual reproduction. Only one stage is being produced, and that is gametocyte stage, which is inside the RBC. Now one more thing simultaneously happens. The Plasmodium which came into our body, my dear children, it is going to feed on the hemoglobin molecule in the RBC. Hemoglobin molecule in the RBC. So while feeding, it breaks the hemoglobin into heme and globin. It breaks the hemoglobin into heme and globin. And what does it do? It is this particular heme which is there, it is very toxic for the organism, which is why it converts the heme to something else which is known as hemozoin, which is very less toxic for them. So heme was toxic, which is why they converted into hemozoin. Now everything is there within the cell. Now when the RBC burst open, my dear children, obviously the gametocytes are free and the hemozoin is also released. The hemozoin is also released in our blood, and hemozoin is toxic for us human beings, which is why we get fevers and chills after every 3 to 4 days. Okay, so hemozoin is a toxin released from Plasmodium infected cell. The questions that can come from malaria is that which stage does it affect? Sporozoite stage. In which stage does it affect? Sporozoite stage. First it goes to its cell, liver cell. Second it goes to its cell, RBC. Where are the gametocytes formed? The gametocytes are being formed in the human RBC. You will also call this as human erythrocytes. Erythrocytes means RBC itself. Where does the Plasmodium undergo sexual reproduction? It happens in the case of mosquito's gut. All these type of questions can come, and I have discussed all of them here, so you need not worry about anything. Done? Cool? So yes, that's all. And one more very important thing, which is the Plasmodium type which causes the malignant form of malaria? It is P. falciparum. P. falciparum. So we have P. vivax, P. malariae, P. falciparum, which causes what? Malaria. In that, the most malignant form of malaria is caused by P. falciparum. I hope that's clear. Yes? So here I have it all. If at all you want to go through it, you can later on. The site where lymphocytes interact with antigens and proliferate to become effector cells are. The sites where lymphocytes interact with antigens and proliferate and become effector cells. What do you mean by that? So basically lymphocytes are type of WBCs, okay, such as B and T lymphocytes. Fine. Now there are two type of organs, lymphoid organs, where these things are going to be produced and they are going to develop. The two type of lymphoid organs are primary, and then the second one is secondary. Primary is where the cells are going to be formed. Okay, they are given birth to. at this particular location, and they mature also, and they also become something that is known as antigen sensitive. They know that there is an antigen, my shatru is there, my enemy is there, this is my shatru, I have to act on it. But it does not start acting here itself. They just become what? Antigen sensitive in the primary lymphoid organs. Examples of primary lymphoid organs, you have bone marrow and thymus. You have bone marrow and the thymus. So bone marrow is a place where all your blood cells are being formed. Now in that, your B cells and T cells are also being formed in the bone marrow. B cells remain here only for becoming matured and for becoming antigen sensitive. The T lymphocytes which are there, they go to the thymus. Thymus is something which is over here. It is behind our rib bone, or you call this as sternum, and it shrinks with age. So thymus is a place where the T lymphocytes are going to become mature and they become antigen sensitive. They are formed here, but they mature at different location, and they become antigen sensitive. Please remember, that is a feature of the primary lymphoid organs. Now when you talk about secondary lymphoid organs, this is where the cells are going to become effector cells. The term effector terms comes over here. That means they are going to show the effect, they are going to do dishum over here. Location, you have spleen, which is just beside our stomach. Spleen is a place where the RBCs, dead RBCs comes and accumulates and things like that. Apart from that, it is richly supplied with these B and T lymphocytes, which are going to trap any organism that comes into the spleen or which is roaming around in our blood. Similarly, we have lymph nodes. Lymph node is a part of your lymphatic system. Just like how we have blood vessels, similarly we have lymphatic vessels, my dear children. They are the ones which will be carrying all the leaked fluids. So our blood vessels are not that great. Sometimes they might leak some fluids. You need to take them back and return it to the heart. Okay, so who does that? It is the lymphatic vessels who does that. Now sometimes what happens, my dear, not sometimes, basically lymphatic vessels will be having certain areas which are known as lymph nodes, and these lymph nodes are richly supplied with again B and T lymphocytes. So any pathogen that comes to the fluid will be caught by them. So lymph node is also coming under this. Now we have MALT. Okay, MALT stands for mucosa associated lymphoid tissue. So mucosa is a layer, an inner layer that is found in our body, the innermost layer. Now whatever lymphoid tissues are associated with it comes under MALT, mucosa associated lymphoid tissue, and almost 50% of the lymphoid tissue comes here itself, or here is a part of MALT. Okay, example, you have Peyer's patch. You have Peyer's patch. Peyer's patch is found in our intestine. In the mucosa layer of our intestine, what do we have? We have Peyer's patch. Tonsils, which is over here. These are also richly supplied with all the cells. This is why we get tonsillitis sometimes, because it is fighting with the antigens out there. So tonsil is also an example. Appendix also is an example. So Peyer's patch, tonsils, appendix, all of them are secondary lymphoid organs. You might be asked to write about all of them as well. Make sure you know to write it. Okay, so site where lymphocytes interact with antigens and proliferate to become effector cells are thymus, the effector cells. So that means you have to talk about secondary lymphoid organs. Thymus is a primary lymphoid organ, which is why this goes wrong. Bone marrow and thymus, both of them are primary lymphoid organs, which is why that also goes wrong. Only Peyer's patch and tonsils are the one which has both of them as secondary lymphoid organs, which is why this is your answer for the question. Cool? Cool. Fine. Now, charas and ganja are the drugs which affect. Charas and ganja, my dear children, they are cannabinoids. Okay, they are cannabinoids, and cannabinoids affect your nervous system? No, ma'am, it comes in between. It affects your cardiovascular system. They are the drugs which are being abused by sports person. I am telling you again, do not forget it. Now, with that, I remember one very important thing. In your book, they are telling you the effect of anabolic steroids. Anabolic steroids. In the case of females and in the case of males. So in the case of male, the testicles becomes reduced, the sperm count becomes less, they will be having mood swings, depression, acne, everything. In the case of female, my dear children, again mood swing and depression is something that is common. Apart from that, they will be having hair growth on their body just like boys. They will be having masculine body and stuff like that. Please go through this. I have explained all this in Brahmastra as well. If at all you feel you need to go and get clarification for a lot of questions out here, you can go ahead with reading the PDF at least of Brahmastra, if at all you do not have time to watch it. Okay? So I've taken questions from Brahmastra itself. So all the questions which I thought might come in FA2 are put over here as well. So a chemical structure is given below. Identify its plant source. This looks like an O. That means it is an opioid. And opioids come from opium poppy, which is Papaver somniferum, which is why your answer is like this. If at all you have something like this also at the end, it would have been a cannabinoid. And the structure is overall different, but yes. Cool? Now, the 3 other 5 markers which also come from the human health and disease chapter are going to be discussed over here. If at all you think that this video is helping you, what do you have to do? Click on the like button right now and subscribe to the channel right away. Do not think twice. Okay? So, this is a question that have been repeated so many times in the model question paper. So, name the causative organism of the following disease. Filariasis. Filariasis is caused by what? Filariasis is caused by Wuchereria bancrofti. Wuchereria bancrofti or Wuchereria malai. Both of them are helminths. Helminths are a type of worms. Okay, they are a type of worms. Ringworm disease, even though it is ringworm, it is not caused by a worm, it is actually a fungal disease. So it takes the shape of a worm, which is why you call this as ringworm disease, but it is actually caused by a group of 3 not virus, fungal species. If I told virus, I'm so sorry. It is caused by fungal species. I'll talk about the name trichophyton, epidermophyton. So trichophyton, epidermophyton are fighting, and then you have microspore, small spore. You can remember like that. Malaria, we know it is caused with the help of plasmodium. You have plasmodium vivax and everything. So here is the term filariasis by Wuchereria bancrofti, or you also have Wuchereria malai. Ringworm, trichophyton. Malaria, plasmodium vivax, P. falciparum and P. malaria. Okay, now since we are solving this question, I want to tell you there are so many diseases given in an NCERT. Please go through each of them carefully. Please know their symptom as well. I can explain a few of them here itself, but I'll still recommend go and clarify everything which is up in your brain. So we have few bacterial diseases, as in these are diseases caused by the bacteria. Typhoid and pneumonia is given in brief, okay, but diphtheria, plague, dysentery, just their name is written, but you should know they are bacterial diseases. I'm so sorry, excuse me. Okay, typhoid, my dear children, is caused by a bacteria whose name is Salmonella typhi. Okay, and what do you see? You have high fever, 39 degree to 40 degrees Celsius high fever would be there, and there will be headache and fever and all of this thing. If it is of high intensity, it can also lead to intestinal perforation and death. The test for typhoid is Vidal test. Now pneumonia is there, my dear children. Pneumonia is caused by 2 bacteria, Streptococcus pneumoniae and Haemophilus influenzae. Haemophilus influenzae is different, Streptococcus pneumoniae is different. Sometimes they will ask you pneumonia is caused by Haemophilus pneumoniae. Both the names are being missed. Please do not be confused. Go through them. Diphtheria, plague and dysentery, they have just mentioned that they are bacterial diseases. So DPD, you can remember like that. Okay, now if you come over here, protozoan diseases are 2. One is malaria and the other one is amoebiasis. Malaria is caused by plasmodium. Amoebiasis is caused by Antamoeba histolytica. It is something that affects your large intestine. It is affecting your large intestine. Over here, typhoid is affecting your small intestine. Please go through all this thing. And whenever I have made a mosquito, that means this mosquito is someone who carries the pathogen. It is not causing the trouble. It is just carrying the pathogen. So it is acting like a vector. Okay. So here, what is the name of the mosquito? The name of the mosquito is Anopheles mosquito. Anopheles mosquito. Please understand, it is a vector and also a host. The only example of being a vector and a host, because here, as I told you, the gametocytes enter and they form gametes, so they have the life cycle going on in Anopheles mosquito as well, which is why it is also a host just like human beings. Okay, for plasmodium. So these are the few things that you should know. In pneumonia, my dear children, the extreme feature that you can see is bluish grey nail and lip color and things like that coming up. Now, helminthic diseases, you have Escheriasis and Filariasis, or you also call this as elephantiasis. Escheriasis, Filariasis and elephantiasis. So Escheriasis is caused by Ascaris lumbricoides. If at all you have a question, what is that particular organism that causes intestinal blockage? Do not think twice. It is this particular thing, that is Ascaris, that causes Escheriasis. Okay, it can also lead to anemia and all those stuff. Fine. Now filariasis and elephantiasis is there, the same thing only. Here elephant like leg is being formed. Why? Because it is going to affect your lymphatic system. So if your lymphatic system is being affected, don't think twice, it is filariasis and elephantiasis. And my dear children, always remember, it is caused by Wuchereria bancrofti. But again, there is a vector, there is a carrier out there. This is again a mosquito. The name of this mosquito is Culex mosquito. It is what? Culex mosquito. So filariasis is transferred by Culex mosquito. Now you have some viral diseases, common cold caused by rhinovirus. caused by rhinovirus. Dengue and chikungunya are also viral diseases, and they are being carried by vectors which are known as Aedes mosquito. Aedes mosquito. So Anopheles for malaria, Aedes for dengue and chikungunya, and Culex for this particular thing. Need to understand, all of them are females. Only the female mosquitoes drink blood. They feed on blood because they are someone who are involved in reproduction. Now, one very important information, with immunization, smallpox disease is completely eradicated. Please know that. Okay, it is completely eradicated. Now, there are some diseases like polio, diphtheria, pneumonia, tetanus, which have been controlled to some extent with the help of vaccines. So these things, go through each of them properly. Have someone in front of you and start asking questions. Make them ask questions and you start answering. That way also you will remember better. Cool? Yes. Now the 5-marker question is differentiate innate and acquired immunity. What is innate immunity? I told you it is present from birth. Acquired, you develop after an infection or vaccination. It is non-specific over here, but it is specific to particular antigens. There is no memory involved in innate immunity because there are no memory cells being formed. But here, B and T cells produce memory cells as well, which is why you have an immunological memory of what has happened. Example is skin and your phagocytes. Phagocytes as in something that will be taking in cell and killing it. For example, you have macrophages. And neutrophils. Which are also known as PMNL, polymorphonucleoleucocyte. Example, antibodies, T cells, or basically an example, B cells and T cells. Okay? Now you have one more question. Mention the cells involved in accomplishing the acquired immunity. What are the cells helping in accomplishing acquired immunity? It is none other than B and the T lymphocytes. B and the T lymphocytes. Nothing else. Acquired immunity, right? Next question. Refer to the given diagram given below and answer the question. Well, this diagram is super important. You should know how to draw it. That is, please practice the diagram of antibody molecule. Now over here, what do you have? You have some question. Name the molecules trapped in the lymph nodes responsible for the activation of lymphocytes that bind to the A and B site. So basically they are telling certain things are trapped in the lymph nodes, and these things which are there, they activate the lymphocytes. So obviously the things that activate lymphocytes are what? Antigens. And they are saying that antigens, basically these things bind at the A and the B site. You know what is the A and the B site over here? Do you know what is A and the B site in the antibody over here? These are known as antigen binding site. Specifically at the top, A and B. So it has to be an antigen which is trapped over here. So name the molecules trapped. What is the name of the molecules? It is the antigens which are being trapped. Now, over here, what does H2L2 in an antibody mean? So over here, my dear children, if at all you see, this is a very long chain of peptide. Similarly, there is a smaller chain of peptide. Two long chains, you call them two heavy chains, which is why you denote it as H2. Two short chains, you call them as two light chains, that is L2. So H2L2 is how your antibody molecule is being formed. At the top, you have antigen binding site. Between two heavy chains, you have so many sulfur bonds, but over here, between heavy and light chain, you just have one sulfide bond. So this structure, please make sure you go through it. It will be super, super important. And in between them also, they have sulfur bonds, okay? Okay, now, again, we discussed that, right? We have discussed this question, we don't need to go ahead. Describe any 3 properties of cancerous cell. Now, cancer is an important disease out there in your NCERT. So, what are the 3 properties of cancerous cell? First thing, what does cancer cell do? They have uncontrolled growth, my dear children. They will keep on dividing. Why do you think that is happening? So, that is one feature, that is uncontrolled growth. But why do you think that is happening? Because usually when two cells they divide and when they come in contact, they show something which is known as contact inhibition. That means they will be inhibiting the growth of other cells when they come in contact. But over here, in this particular case, my dear children, the cancerous cells lose this ability. So which is why they will keep on dividing even when they come in contact. So first feature is uncontrolled cell growth. But why is it happening? Because they have lost contact inhibition. Apart from that, you might have seen the cancer cells, we say that even though we have removed from here, they have traveled to other places in our body. You call this feature as metastasis. So metastasis is also one feature that you need to know. So uncontrolled growth, they divide rapidly without responding to normal regulatory signals. Loss of contact inhibition, they grow even when in contact with other cells. Okay, and metastasis, they can spread to distant body parts via the blood or the lymph. So these are the three features that you need to write. Okay. Now, name the group of genes that have been identified in a normal cells that could lead to cancer. So they are saying that in normal cells there are some genes that can lead to cancer. What is the name of this gene? What is the name of this particular gene? You call them as proto-oncogene. What do you call them? You call them as proto-oncogene. And why do you think they can cause cancer? So usually what they do is that they do the jugular function of forming some proteins. But suppose there are some mutations happening up over here, some changes in the DNA happening over here, they will turn a normal cell to what? Cancerous cell, which is why you call them proto-onco. Onco is for cancer, proto-oncogenes. Okay, and you might have heard about some viruses also, oncogenic virus. You know what they do? They have the genes of causing cancer, they transfer it to our cells. That is, oncogenic genes are being transferred to our cells. Anyways, so these are the normal cells that we have, but if at all there is any mutation, it can lead to cancer cells. So name the group of cells, group of genes that have been identified. It will be proto-oncogenes. Now, which techniques are useful in detecting cancer of internal organs? Internal organs, my dear children. So basically, if it is internal organ, it has to be specifically, my dear children, CT scan, computer tomography. But again, we have to talk a bit about all the other things because it's a 3-marker question. So we'll talk about all the cancer detecting mechanism. First of all, you have biopsy and histopathology. That means suppose you have a growth inside your body. You cut some part of it. You call this as biopsy. Now when you put it under testing, you call this as histopathology. So biopsy and histopathology, microscopic examination of tissue to detect malignant form of cancer. Now CT scan is there. Okay, computerized tomography method, my dear children, computer tomography method, where what do? You put X-rays. You use X-rays to give you a 3-dimensional image of the internal organs, okay, which will be helping in detecting the cancer or tumor in the internal organs. So CT scan or computer tomography is being done using X-rays, ionizing radiation. Remember, X-rays are not safe. They're ionizing radiation, okay, and they lead to generation of 3D structure of the internal organs. So we'll get to know things better. Now, there is another method, that is magnetic resonance imaging, MRI. Over here, you are not going to use X-rays, rather you are going to use magnetic fields and also non-ionizing radiations. That means this is still safe. Why? It is being used to understand about the living tissues, to detect any problem in the living tissues. So if at all this question comes, that which is that technique used to understand any problem is there in the living tissue, it is MRI. For internal organs, it is CT scan. Apart from that, there is radiography things also, such as you directly use X-rays and try to, normal X-ray things which are there, that can be also used. Okay, apart from that, cancer people, my dear children, they produce specific type of antibodies. So if at all I know that these are antibodies produced only in cancer patients, and I take some sample from someone and I see the same antibody there also, I can be very sure that that person also has cancer. So antibodies specific to cancer, testing of antibodies specific to cancer, that can also be done. Now, why are cancer patients often given alpha interferons as a treatment? I told you, alpha interferons is a biological response modifier. It is for increasing your immunity. That is why you are given alpha interferons. You might be asked, what are the different type of things that causes cancer? So you call them as carcinogen. You have physical carcinogens such as X-rays, gamma rays, UV rays, things like that. You have chemical carcinogens, as in chemicals that can cause problem. For example, tobacco smoke, it can lead to lung cancer. Then you have biological carcinogens such as proto-oncogenes, which is there in our cell, and oncogenic viruses, which transfers their oncogenes into our body and causes cancer. Okay? Please be sure about all these things which come. What is it? Have I again copied the same question? That is why I saw that. Okay, anyways. Question number 17. Give the schematic representation of replication of a retrovirus. Now this is a super important question. You might be asked to draw this as well. So listen to me. HIV is a retrovirus. Agreed? HIV is a retrovirus. This retrovirus, my dear children, will be having RNA as the genetic material. They do not have DNA, they have RNA as the genetic material, and both this RNA is put inside a protein covering which you call as capsid. Okay, which you call as capsid. Now basically it is coming to affect our cells. So the retrovirus which is there, it comes to infect normal cell. It has come over here. What does it do? It transfers its RNA inside. Remember, both this RNA are single-stranded. They are not fused together. They are single. They transfer the RNA inside. So viral RNA is introduced into the cell. Now what happens? With the virus, they have some enzymes also with them. They have some protein structures with them. You call them enzymes. One such enzyme is reverse transcriptase. Reverse transcriptase. What does it do? It reverses the process of transcription. You know how usually transcription happens? So in transcription, from the DNA, you make the RNA. Now when you are doing the reverse of it, what do you call? Reverse transcription. When you are doing the reverse of it, you will call it reverse transcription, my dear children. Okay, so here you need an enzyme, and that is reverse transcriptase. So according to the central dogma that we have studied, from the DNA we always get the RNA, from the RNA we get the protein. This is the central dogma. Anything that deviates the central dogma is happening over here in the form of reverse transcription. So why do you think it is forming the DNA? Because our genome has the DNA, and if at all it wants to be a part of our genome, it has to form its own DNA. Now the viral DNA incorporates into the host genome. It will come and get incorporated into our genome. And what will happen? Basically it will use our machinery to form RNA, to form other proteins, to form the covering, everything. And that's how you get new viruses being made. Now what can be happening is that they can give you a flow chart, they can make boxes and can ask you fill in the box. So you should know, virus comes to attack a cell, they insert their genetic material which is RNA, they reverse transcribe it with the enzyme reverse transcriptase. They form the DNA now, viral DNA. The viral DNA gets accumulated into the host or incorporated into the host genome. Now they use the host genome's machinery for production of viral RNA, viral proteins and everything. And together you get new viruses being produced and they go on infecting the other cells. This picture is important, the flow is important, please know all of it. And with that, I think we have completed the chapter Human Health and Diseases, important questions. One very important thing that I want to tell you right over here is that please remember the first hepatitis B vaccine. Hepatitis B vaccine was made with the help of yeast. Please remember, that's an out-of-box question, but it came to my mind, I just told you. Okay, and one more thing. So hepatitis B vaccine was produced with the help of yeast. And my dear children, one more thing, in the case of allergy, what is happening? You have a cell's involvement, you call this cell as mast cell. On the mast cell, I told you which antibody will be there in allergy, IgE. So these antibodies are binding. Now suppose some allergens comes and bind over here, it will indicate the mast cells to release two very important things. One is histamine and the other one is serotonin. Histamine and serotonin is released by the mast cells. Now what will happen? It will lead to vasodilation. That means blood vessels are going to get dilated, which is why we get swelling and things like that happening. Now to treat allergies, what do you have? You take antihistamines, something that will work opposite to the histamines. So you take antihistamines. Apart from antihistamines, you also depend on this thing. What do you call? You also depend on steroids. Okay, you also depend on steroids. Please remember that. You also depend on steroids and also adrenaline. So antihistamine, steroids and also adrenaline was the one that were being used, is what I remember. Okay, to treat them. That's all. Fine. So microbes in human welfare, let's begin this particular chapter. I hope, my dear children, you are feeling somewhat confident out there. I am discussing all these things for you guys. Make sure you are there with me throughout the session. Okay? So, Saccharomyces cerevisiae, commonly called brewer's yeast, is used for fermenting what? Saccharomyces cerevisiae is yeast. You call it brewer's yeast when you use in brewery industry, for example, formation of some alcoholic drugs and stuff like that. So not drugs, alcoholic drinks, okay? Now you call it baker's yeast when it is used for baking. So anyway, it is all in all. Now here you are using in brewer industry for fermenting malted what? What are malted? First of all, malted means, for example, I have some pulse or cereal out there. It has started undergoing germination process. And then we stopped all the condition that will make it undergo germination anymore further. It will stop the process. So now in this, what do you call, halfway there, okay, in this condition where it has completed, somewhat started off germination but has not completed it, you call this condition as malted condition. And over here, you are going to use malted cereals for the process of fermentation. Why? Because as I said, when germination is being started off, what will happen? The starch which is being stored is being broken down to simple sugars, and that is exactly what we wanted. Because now the fermentation process would be even more easier. If at all it was in the form of starch only, it would have been taken longer time. But now it is easier because the process of germination had started, but now we have stopped it. So malted cereals are used. Malted cereals and fruit juices are used in brewery industry for formation of things. Okay. Now, because of the presence of which of the following vitamin, curd is considered as nutritionally richer than milk? Why do you think curd is considered nutritionally richer than milk? Because, my dear children, curd has a very, very important thing which is known as vitamin B12. So, what happens is that basically in the milk when we add lactic acid bacteria, it produces lactic acid. And this lactic acid basically works against the milk protein, which is casein. It partially breaks down the casein, which is why you get clumps of things out there. And apart from that, this particular lactic acid bacteria also releases vitamin B12. It releases for its own purpose, for its own cell division and stuff like that. But over here, anyway, it is mixed in the curd. So now we are going to take the most use of it. So vitamin B12 is the answer for this question. Produced by lactic acid bacteria when it is converting milk to curd. Okay? Mycorrhiza is involved in absorption of dash from the soil and passes it to the plant. What is mycorrhiza? So mycorrhiza, my dear children, always remember it is a combination of fungi plus the root of the plant. What does this fungi do? How does it help? This is symbiotic association. That means both has to help each other. How does this fungi help? Basically, roots can go deep, but in the case of fungi, they will also have rhizoids that thing. They can go even more deeper, and that is how it basically helps in the absorption of one very important thing, which is phosphorus. Don't go and write potassium. It is phosphorus. So rust is getting from the soil, you can remember like that. It is phosphorus. Apart from that, the fungi also provide resistance, okay, or tolerance, resistance to pests or to pathogens, which basically can try and damage the roots. Apart from that, tolerance to what? Salinity. There is too much amount of salt in the soil, still the plants would be able to survive with the help of this particular fungi. Salinity, if there is no much water, still they can survive because of this particular fungi. Okay, so phosphorus absorption, resistance to pathogens, tolerance to salinity and drought, and also they help in overall growth and development. So ma'am, when fungi does all this thing, what does the root of the plant do? Basically, plants are responsible for photosynthesis. So they do photosynthesis, they provide the fungi with organic materials or the organic matter, which allows the fungi to stay active. Okay, so this combination is known as mycorrhiza, and since it is providing a very important nutrient from the soil, this is also an example of biofertilizers. So fertilizers are basically chemicals which provide nutrition to the soil, right? Biofertilizers means organisms which act like fertilizers, organisms which basically helping in increasing the nutritional quality of the soil. One such example is mycorrhiza, and please remember this fungi belongs to glomus. This fungi belongs to glomus. So answer for this question is B. Now, during sewage treatment, the gas produced in anaerobic sludge digester include. So whenever gas is being produced, my dear children, please understand, and that to sewage treatment, then it has to be biogas. And what does biogas contain? first thing that is containing it contains is methane, which is shown by CH4. Don't go and write CH3 or don't go and tick the option that has CH3. It is CH4. Apart from methane, you have H2S, hydrogen sulfide. Okay, apart from that, you have CO2, carbon dioxide. Now, if at all an option has 3 of these things, you go with that. If at all it is not H2S, rather it is given as H2, but here methane and CO2 is given, still the option is correct. Please go with the other option. So over here, hydrogen sulfide, nitrogen is given wrong. Methane, oxygen is not at all there. Methane, hydrogen sulfide and carbon dioxide. Methane, hydrogen sulfide and carbon dioxide, 3 of them are there, which is why this is my answer. Okay, here sulfur oxide is there, that won't be the answer. Always remember, biogas is produced with the help of anaerobic bacteria. Bacteria that does not need oxygen. They are the one that is responsible for the release of biogas. Always remember that. So yes, either this or this. Chalo. Propionibacterium shermani is used for the production of Propionibacterium shermani. I always tell like this. So Shermani, why it is getting shy? Because my dear children, over here, this particular bacterium is basically releasing CO2. And whenever it releases CO2, the path gets what? A hole. The path gets a hole. And this type of thing that it is producing is known as Swiss cheese. Always remember. So Propionibacterium shermani is used for the production of Swiss cheese as it releases CO2. Fine. Don't forget. SS. You can remember like that. Roquefort cheese is not produced by this. I think I have unknowingly marked Roquefort, but this is not the one. This is the one. Please go through it, correct it. Okay. How is Roquefort cheese produced? With the help of another type of fungus, actually, not a bacteria. It is Penicillium roqueforti, not Penicillium notatum. Okay, that is there for production of penicillin. This is some other fungus, which is Penicillium roqueforti. You don't need to study about that, just understand it's a fungus. This is produced by a fungus, and this is what? A bacteria. Okay. Now, Alexander Fleming discovered penicillin while working on. So, we just discussed about that. Penicillin, my dear children, it is the first drug being produced. First drug, or basically antibiotic, medicine, antibiotic being produced. You know how it was produced? So, basically, Alexander Fleming was working on some bacteria whose name is Staphylococci. Staphylococci. Okay, now one day he forgot to wash the dish and he left. The next day he was worried, oh my God, the culture would have grown so much. But then he came to know that basically a fungus mold is being developed over here. A mold is being developed over here. I was like, okay, mold is developed, so what? But because of the presence of this mold, the bacteria were not able to grow. That was kind of fascinating. Then he got to understand that probably this mold was releasing something that does not allow the bacteria to grow. So what is this something? That has to be antibiotic. It has to be antibiotic. What is the name of this particular fungi which was growing? It was Penicillium notatum. Penicillium notatum. So you can remember, does not allow the growth of bacteria. Penicillium notatum does not allow the growth of bacteria. What is the bacteria? Staphylococci. Staphylococci. Okay, it is not streptococcus. Always remember, that is a usual difference. Cocci is anyway there. L is here, L is there also. Okay, so it is Staphylococci. Remember it. So it was discovered by Alexander Fleming. The name of the antibiotic was penicillin, and later on few more people came in, that is Ernst as well as Howard Florey. They also came into the picture, and together they brought up this particular thing, that is we can use this particular antibiotic out there, and they used it in World War II. In World War II, this particular antibiotic was used to treat the wounded soldiers. My dear children, this is Alexander Fleming, and 3 of them, they got the Nobel Prize in the year 1945. 3 of them together got the Nobel Prize in the year 1945. So Alexander Fleming was the person who discovered. Later it was developed by Ernst Chain and Howard Florey. 3 of them got the prize. Cool? Chalo. Which one of the following alcoholic drinks is produced without distillation? What do you mean by distillation? Distillation is where you make the water get evaporated. That means the amount of alcohol is going to be concentrated because the water has gone in the form of evaporation. Concentrated alcohol is found in where? Whiskey, rum, brandy. Whereas when you talk about wine, when you talk about beer, the amount of alcohol is less. The content of alcohol, concentrated alcohol is less because they do not undergo distillation. So here you have been asked which one of the following alcoholic drinks is produced without distillation. So without distillation it will be what? It will be wine. With distillation it are these two things. So it will be wine or it can be also beer. It can be wine or the beer. Got it? Yes. Just a second, okay? Yeah. Now, which of the following is not an advantage provided by mycorrhiza to the plants? Which is not an advantage provided by mycorrhiza to the plants? Now, this is very important because I told you what is the advantage that is provided by mycorrhiza. So, resistance to root-borne pathogens? Yes, ma'am. Tolerance to salinity and drought? Yes, ma'am. Overall increase in growth and development? Yes, ma'am. But what about increase in yield quality? Yield means their production is more now. No, that is not controlled by mycorrhiza. So basically this is the odd one out. Please remember it. Increase in yield quality is the odd one out. Now, the microbial biocontrol agent for butterfly caterpillar is, suppose you want to kill the butterfly caterpillar. What is the biocontrol agent? So usually we have pesticides, but pesticides make the use of chemicals, and suppose we do not want chemicals to be used up over here. We do not want chemicals to be used up over here. So we depend on other organisms that probably can kill this particular pathogen. So we say dushman ka dushman, right? So similarly over here, this particular agent, this particular organism is going to feed or basically destroy the butterfly caterpillar, which is going to affect the plant. Now you have been asked what is that biocontrol agent. Since you have been asked about butterfly, my dear children, your answer for this question is going to be Bacillus thuringiensis. Not this, not this, not this. What is Bacillus thuringiensis? We have studied in biotechnology. At least some of us have studied. If you do not know, it's okay. So Bacillus thuringiensis is a bacteria. Now this particular bacteria, my dear children, it produces a toxin but in the inactive form. It produces a toxin in the inactive form. You call this as prototoxin. It is inactive, please remember. Now, what happened? Basically, suppose a butterfly caterpillar came in and ate this bacteria. Somehow had got this bacteria inside its body. Probably this bacteria might be on some leaf and stuff like that. Now this bacteria is inside the gut. Is inside the gut of what? Of this organism. Now when it is inside the gut of this organism, what will happen, my dear children? Does anything happen to the inactive toxin out there? Yes. It gets the condition for becoming active. Here in the gut of the organism, you have alkaline pH. There is a basic pH, which is why the inactive toxin which is there, it will become active in nature. It becomes active in nature. And when it is active, what will happen? There will be perforation, there will be pores being formed in the epithelial layer of the midgut region. So which gut? Midgut. In the midgut region, there is a layer of epithelial cells. These epithelial cells, my dear children, they will be having small little pores because of the active toxin. So that is the thing. Now, what do you do to basically get this bacteria inside this organism? So one thing that can be done is that you get the spores of this particular bacteria, you can mix it with water and spray it on the plant. Any organism which comes and eats it will be dying. Apart from that, you can do genetic modification. You can take the gene that is producing this inactive toxin and put it inside the plant, so that plant is also producing the same toxin. That is how you get BT cotton, BT brinjal, because you have the gene being put. What is the gene over here? It is known as cry gene. It is known as cry gene. You will be studying about it in the next chapter, that is biotechnology, principles and processes. But anyway, so microbial biocontrol agent for butterfly caterpillar is Bacillus thuringiensis. It is also the biocontrol agent for other insects and also for brassicas, for example, cabbage, cauliflower and stuff like that. Okay? Now select the correct statement from the following. Biogas is produced by the activity of aerobic bacteria on animal waste. I always tell you, it's never aerobic. Aerobic means they need oxygen. Biogas is produced in the absence of oxygen. That means you are talking about anaerobic. You should be talking about anaerobic, but here it is aerobic, which is why this is wrong. Next, Methanobacterium is an aerobic bacterium found in the rumen of cattle. Methanobacterium is again something which is going to sustain in anaerobic medium. Okay, anaerobic means there is no oxygen. So it is found in the cow dung, and the cow dung is used for formation of biogas. Why? Because it has this bacteria which can work in anaerobic conditions. So it is not an aerobic bacteria, it is anaerobic bacteria. It is actually found in the rumen of the cattle. Because in the rumen of the cattle, the plant is being eaten by the cattle, right? And the plant has cellulose, my dear children. For breaking down cellulose, you need certain bacteria like that, and is also responsible for production of biogas. Now, biogas, commonly called as gobar gas, is pure methane. Is it pure methane? We studied it's a combination of three things: either methane, H2S and CO2, or methane, H2 and CO2. Not pure methane. So this is also wrong. Now, activated sludge in settlement tanks for STP is a rich source of aerobic bacteria. Ma'am, we did not know this answer now because we have not studied sewage treatments. Do not worry, I'll teach you within a few time itself. But until then, please understand that this is correct. So, three options are wrong, so that means the other one is correct. Activated sludge in settlement tanks of STP, STP means sewage treatment plants, is a rich source of aerobic bacteria is being said. Is it true or false? Let's talk about that here itself. Since I do not want any doubts to be coming, anyway, this is the last, MCQ. So, we'll directly go to this question, the sewage treatment. Okay, because the sewage treatment question is super important. Initially, they were being asked in the form of 5 markers, but now this particular chapter does not give you 5 marker questions. It can be either 2 marker or 3 marker questions that come from this chapter. So you can expect they can ask you what is primary treatment for a 2 marker. It has been coming in the model question paper this year. So you should know all the steps. It can be either of them can come in a 2 marker or a 3 marker question format. So first thing, whenever you have sewage, the first thing that it undergoes is known as primary treatment. And what do you mean by primary treatment? In the case of primary treatment, my dear children, things are going to be filtered out, as in the things that can be floating, they go at the top. The things that are of heavy weight, they will be sinking towards the bottom. You will call this as the sediment. The things that float will be known as what? Okay, will be known as floating debris. Anyways, so things that are floating out there, I can remove it with the process of filtration. This is the first step of primary treatment. The things which are settling down over here, I can remove them with the help of sedimentation. Now, what is being left over here, my dear children? What do you call this? You call this as the primary effluent. What happens to the primary effluent? This is being taken for the next treatment, that is secondary treatment. And what do you call this thing which is getting accumulated over here? You call this, you have a specific name, you call this as primary sludge. Ma'am, where is this all happening? What do you call this tank as? This particular tank which is there, you call this as a settling tank. So in first condition, in primary treatment, you use a settling tank where you use the process of filtration to separate out the floating debris. You use the process of sedimentation to remove the sedimented things. The sedimented things are known as primary sludge. The thing that we are going to take ahead for secondary treatment is known as primary effluent. It is all being done in the settling tank. Next thing, you are going to do the secondary treatment. How does it work? Let's go ahead. So first, let me read it. Involves the physical removal of particles, large and small, from the sewage through filtration and sedimentation. Initially, floating debris is removed by sequential filtration, by filtering it out. The grit, soil and the small pebbles are removed by sedimentation. All the solids that settle from the primary sludge and the supernatant forms the effluent. This is known as the effluent. The effluents are from the primary settling tank taken for secondary treatment. They are taken for secondary treatment. Okay, the primary effluent is taken to secondary treatment. Now, what exactly happens in the secondary treatment? So, for that, we need to draw some diagram. So the secondary treatment which is there, so first treatment was done in a settling tank. So, filtration was done, sedimentation was done. This was the primary effluent, right? This you put inside an aeration tank. You put it inside the aeration tank. That means you're going to provide air in the form of oxygen and stuff like that. Now the primary effluent is there. What happens, you know what? There might be certain aerobic bacteria over here. What do you have? You have some aerobic bacteria over here. That means the ones that will require oxygen. And since it is the primary effluent, my dear children, what will be happening? There will be organic matter also. Organic matter as in waste content which has to be broken down. Aerobic bacteria is there. You are putting it in aeration tank. Aerobic bacteria, they will increase their number. They will definitely increase their number, and they will team up. They will team up with some fungal filaments. So aerobic bacteria plus some fungal filaments, you will now together call them as flocks, which is super important. Flocks. This flocks which is there, which is a combination of both of them, is going to act on the organic matter and break it down. And is going to break it down. Now you only tell me, this is aerobic bacteria. It wants to break down the flocks which is there. It wants to break down the organic matter. Does it need oxygen for this? Obviously, ma'am, you need oxygen, which is why we say the biochemical oxygen demand. Okay, the demand for oxygen, the biochemical oxygen demand is going to be super high. It is going to be super high. Now, once the organic matter, my dear children, is reduced, the organic matter, when it is reduced, do you need the same amount of oxygen now? No, ma'am, which is why we say the BOD is less. BOD goes less because the oxygen demand has become lesser when organic matter is not there. You get it? So you have to work until the BOD is less. Or you can be asked this question, how does BOD help in understanding the pollution level? If there is high BOD, that means you need more aerobic bacteria, or those aerobic bacteria need more oxygen to break down the organic compound of them. If there is less pollution, there is less need of oxygen. You get it? So that is how biochemical oxygen demand is going to help as a factor. Oxygen demand. And flocks is a combination of aerobic bacteria as well as the fungal filaments. This is happening in aeration tank. Slowly what will happen, my dear children? The organic matter which is there, it is being broken down completely. The organic matter which is there, it is broken down completing. Everything is cool. So what do? We put it inside the next settling tank. Again, we are using what? Settling tank. Now, in the settling tank, what will happen? The fungal filaments plus the aerobic bacteria, that means you're talking about the flock. They are going to settle down. They are going to settle down, and this is what you call as activated sludge. What do you call this as? Activated sludge. Now they have settled down. Whatever is left behind is nice water, is purified thing that we have got. This thing that we have got is sent to rivers, Now what do we do with the the settled activated sludge out there? My dear children, we have to make sure it is also gone because it is of no use for now. So we will take some part of it. Okay, we'll take some part of it and we will put it in the aeration tank such that the next primary effluent when it comes, it has little bit of aerobic bacteria. From here you got the aerobic bacteria, right? So this is going to act like an inoculum as a base so that aerobic bacteria can still form. Whatever is left behind, the activated sludge which is left behind is been put to the next thing which is known as the anaerobic sludge digester. Easy. Do not mug anything up. Anaerobic sludge digester means in anaerobic condition without the help of oxygen, it is going to digest this particular sludge. This sludge contains what? Aerobic bacteria. This sludge, this was a question that was being asked there. So activated sludge contains aerobic bacteria. So is it true? Yes, it contains aerobic bacteria as well as the fungal filaments. Okay. Now this is put in anaerobic sludge digester where the anaerobic bacteria, the bacteria that does not need oxygen, will be acting on this aerobic bacteria as well as the fungal filaments and breaking it down and producing the gas which is biogas. Anaerobic bacteria, that means it is going to produce biogas. In biogas you have methane, H2S as well as carbon dioxide. So with the sewage you got the proper form of water. Apart from that, basically you are generating biogas as well. The question which is being asked is that activated sludge, where is it going to settle down? In the settling tank. Okay, the primary treatment, where it is done? Settling tank. Aeration tank is for what? This particular thing. Secondary treatment is started off here. What is flocks? Why is flock important? Because they are the ones that is going to work or break down the organic matter into simpler compounds. What is the use of BOD? I told you if there is high BOD, it shows there is high pollution as well. Because high BOD means high oxygen demand by the aerobic bacteria to break more amount of organic matter. That means high pollution. You can make arrow and show. Okay, so you work until the BOD goes less. And now put it in the settling tank, everything gets settled down. This is known as activated sludge. It contains aerobic bacteria. Again, you put it inside anaerobic sludge digester to digest it and you get the biogas. Simple. This is your sewage treatment. Take a screenshot because this slide might go. I have added this slide now. So, we were here. The primary effluent is passed into large aeration tanks. This allows vigorous growth of useful aerobic microbes into flocks. The growth of microbes consumes the major part of organic matter in the effluent. This significantly reduces the BOD. BOD refers to the amount of oxygen, this is also important, amount of oxygen required to oxidize total organic matter by bacteria present in 1 liter of water. In 1 liter of water. BOD is the measure of organic matter present in the water. Greater the BOD of wastewater, more is its polluting potential. Once the BOD of sewage reduces, the effluent is dispersed into the settling tank where the bacterial flocks are allowed to sediment. This sediment is called activated sludge. Small part of the activated sludge is pumped back as an inoculum to the aeration tank. The remaining sludge is pumped into anaerobic sludge digester. In the anaerobic sludge digester, there is other kinds of bacteria which grow anaerobically, digest the bacteria and fungi in the sludge, producing what? Biogas. That's all. Fine. I hope this is clear. And now, this particular question which was there, it also might be clear. Activated sludge in settlement tanks of STPs is a rich source of aerobic bacteria. Now, 2-3 marker questions which are there. One question we sorted it. Now discuss the role of fungi as biofertilizers. This is also something that we have discussed. Fungi as a biofertilizer, for example, mycorrhiza, which belongs to glomus. So glomus lives in close association with the root of the higher plants, enhances the phosphorus absorption from the soil and transfers it to the plant, increases resistance and tolerance, resistance to pathogens and tolerance to salinity and tolerance to drought. Overall increase in plant growth and development is done by them. Mutual benefit, plant also gives them organic matter. There is no increase in yield. No increase in yield because of fungi. Other biofertilizers also you should know. So there are some nitrogen fixing bacteria. There are some other bacteria also which comes under cyanobacteria. That means the one that can prepare their own food. Remember, they are put in different category. If at all you are being given option this, choose the nitrogen fixing bacteria. Go for this. What are the examples? Rhizobium, Azospirillum, Azotobacter. Rhizobium, Azospirillum, Azotobacter, they are the nitrogen fixing bacteria. Rhizobium is found in leguminous plants such as pea plant and still things like that. Azospirillum, Azotobacter, they are also free living bacteria that enrich. They are free living, they are not associated with any plant, but they basically fixes the nitrogen, as in nitrogen is in the form of N2, right, which has 3 bonds. It is very difficult for the plants to break these bonds and absorb it, which is why other organism breaks it down, converts into something else and give it to the plant. That is known as nitrogen fixation. For that you have Rhizobium, Azospirillum, Azotobacter. Rhizobium is found in the root nodules, whereas Azospirillum and Azotobacter, they are individual, they are separate, free living ones. Mycorrhizal fungi, we just saw them. Now we have cyanobacteria, the bacteria that can produce their own food. Example, you have 3: Anabena, Nostoc, Oscillatoria. Anabena, Nostoc, Oscillatoria. Instead of ano. Okay, instead of it is ano. Anabena, Nostoc, Oscillatoria, they are cyanobacteria which can prepare their own food. That means they are autotrophic. And my dear children, they also fix nitrogen. They also increase the soil fertility. And they are used as biofertilizers in paddy fields. In what? Paddy fields. Okay, they increase the organic matter content. So bacteria is Rhizobium, Azotobacter, Azospirillum. Cyanobacteria is Anabena, Nostoc, Oscillatoria, and all of this is the example for mycorrhizal fungi. They are biofertilizers. So if at all you are asked, explain the role of microbes as biofertilizers, this is your answer. If at all you are asked, name the microbes that help in the production of following bioactive agents, please understand this question is super important. This has come in 3 model question papers of this year. So you have to tell that they are bioactive agents. Bioactive agents means they are being used for our own purposes, but you get it from microbes. So cyclosporin A, my dear children, you get them from a fungi. You get it from a fungi The name of the fungi is Trichoderma polysporum. You have to name them, that is the question. Okay, so it can be named the fungi and also the function. So cyclosporin A is you get it from Trichoderma polysporum, and it is used as an immunosuppressive drug. For example, I have taken a kidney from someone else because my kidney were not working. My body will react to it because it's a foreign particle. But now I do not want my body to react so much, which is why I take immunosuppressants. So cyclosporin A is an immunosuppressant. You get it from a fungi that is Trichoderma polysporum. I know it might be too much for you to remember so many names, but trust me, whenever you go ahead, read, it becomes all the more settled in your brain. That's how even I learned. Okay, now streptokinase, you get it from streptococcus, not staphylococci, staphylococci penicillium. Streptococcus. Streptokinase is what? It's a clot buster. It is a clot buster. That means any clots which are being formed in the body, it has to burst it. That is how we can be preventing heart attack or what you call as myocardial infarction. Now we have one more thing, statins. Statin, my dear children, stabilizes the blood cholesterol level, or you can say it lowers the blood cholesterol level. You get it from Monascus purpureus. Where do you get it from? Monascus purpureus. So there three names are there. I want you to just read it once, pause the video, read them once, and then close your eyes and just read out their names. Streptokinase. Okay, you get it from Streptococcus. It is a clot buster. Cyclosporin A, you get it from Trichoderma polysporum. It is what? It is a immunosuppressive drug. Statin stabilizes blood cholesterol level. You get it from Monascus purpureus. Okay, please go through it. They are bioactive agents. Now you have other things also. Explain the role of microbes in industrial products. Now over here, industrial products come under them also they come. Okay, penicillin, notatum penicillin, streptococcus, streptokinase, all of them. Apart from that, you have few of them here also. And you might get matched the following question from here. So where do you get citric acid from? From Aspergillus niger. From Aspergillus niger. This is a fungi. Where do you get acetic acid from? You get it from Acetobacter aceti. Acetobacter aceti. Acetic acid, Acetobacter aceti. Names now sound similar. So you can do match the following question, but you should know even if they are asking you to write the names. Okay. Butyric acid you get from Clostridium butylicum. Clostridium butylicum. Lactic acid you get it from Lactobacillus. So three of them basically they are bacteria. Three of them they are bacteria. What about ethanol? Ethanol, which is basically form of alcohol, my dear. Where do you get it from? With the help of yeast, which is Saccharomyces cerevisiae, which is again a fungi. So children, read it out. Citric acid, where do you get it from? Aspergillus niger, ma'am, it is a fungi. Acetic acid, Acetobacter aceti. Butyric acid, Clostridium butylicum. Lactic acid, Lactobacillus. Ethanol, Saccharomyces cerevisiae. That is again a fungi. Two fungi, three bacteria. Okay. So if at all you are asked what is the fungi that acts in what do you call production of industrial products, you should know that these are the examples. These two are the examples for fungi, these two for bacteria and all of them. This is also, and Penicillium notatum is also a fungus. Trichoderma is also a fungus. Anyways, so this is for industrial products. Now we have one more thing. Explain the role of microbes in household food products. So we saw them as biofertilizers, we saw them as industrial products. Now in household functions. So lactic acid bacteria, which turns the milk into curd. Okay, it is found in household conditions, right? It partially digests the milk protein, the casein. Milk protein is casein. It partially digests it, which is why we have small clumps being formed, and it makes the curd rich in vitamin B12. Baker's yeast, Saccharomyces cerevisiae, this is also known as brewer's yeast. Here baker's yeast because it is helping in the formation of bread. Toddy is a traditional drink which is made by fermentation of the palm plants out there. Okay. Now, large holes in the Swiss cheese are due to production of large amount of CO2 from Propionibacterium shermani. And there is Roquefort cheese, which is ripened by specific fungi, which is Penicillium roqueforti. The name is not given in NCERT. You don't need to remember that, but at least remember it's a fungi. So, lactic acid bacteria, baker's yeast, toddy, Swiss cheese, Propionibacterium shermani, and then Roquefort cheese by fungi. They are for household products. Cool. Now, So I know that it's too much for you to, if at all this it is being watched by someone who have not read the chapter once, it might be difficult for you, but I request you to please, pause the video and try to remember it. Okay, make your own thing, remember, actively recall and write. Do not see it and write it, try to actively recall and write it. It will be very, very helpful for you. Explain the role of microbes as biocontrol agents. Biocontrol agents, as I told you, Bacillus thuringiensis, it was a biocontrol agent, right? Similarly, you have few more things. For example, you have some insects which are going to kill other insects, such as ladybird beetle. You might have seen red and black spotted beetles out there. They feed on aphids. They feed on aphids, which are crop pest. Now you have dragonflies. They feed on mosquito larvae. So ladybird beetles, they feed on aphids. Dragonflies, they feed on mosquito larvae. So these are predatory insects. 2 examples done. Microbial biocontrol agent, you have Bacillus thuringiensis, as I told you. It can kill butterfly caterpillars. It is available as dried spots in sachets mixed with water and sprayed on plants like brassicas, cabbage and cauliflower, and also fruit trees. Now fungal biocontrol agent is there, trichoderma. Ma'am, I remember this name, trichoderma. Yes, trichoderma polysporum, which gave cyclosporin A, that is a immunosuppressant. Here this is different. The trichoderma fungi here basically does not allow other fungi to damage any roots and things like that. So they are found in the root ecosystem, does not allow any other pathogen to grow out there. So it is also acting like a fungal biocontrol agent. So predatory insect, ladybird beetle, feeding on aphids, dragonflies feeding on mosquito larvae, and microbial agents you have Bacillus thuringiensis, fungal agents you have trichoderma, which is in the root ecosystem. Now we have one more, viral biocontrol agents, and for that you have baculoviruses. You have baculoviruses which come under nucleopolyhedrovirus. Please remember the name, it's important. Baculovirus, they come under nucleopolyhedroviruses. They infect only specific insect pests and also arthropods. Specific insect pests and also other arthropods. Insects are also arthropods. Anyways, eco-friendly, does not harm plants, mammals or anything else. So baculoviruses, or they come under nucleopolyhedroviruses, and other examples, please remember them. They are all biocontrol agents. Okay? I hope this is crystal clear. So it is super important. Go through this. I know it's might be difficult for you, but you think it's difficult. It's not as difficult as you think once you start trying. Okay, so it's my request to you. Pause the video and try to solve it by yourself as well. Now we move on to the next chapter, which is biotechnology, principles and processes. If you want to take a break, you can take a break. I'll also be right back. And my dear children, if you feel that you cannot do it, let me tell you, you are halfway there. It's just half more portion out there, not half more, even a little bit portion also remaining out there. You will be able to do it. Okay, keep going. Okay children, I hope the break is done and we are all energetic now, right? So let's get into the next chapter, that is biotechnology, principles and processes. We'll be discussing the 1 markers first and then the detailed long and short answers. First of all, if at all you like the video and you have not clicked on the like button yet, do that and share it with your friends. Do not forget to subscribe to this channel. Chalo, 34th question. In the year 1963, the 2 enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. What is happening? So in E. coli, which is a bacterium, there were some viruses attacking them. What is the name of the viruses? It is bacteriophages. So bacteriophage were attacking them, and in response to that attack, E. coli basically was producing 2 very important enzymes. Okay, was producing 2 very important enzymes. So E. coli were one of them cut the DNA and the other added dash group. One of them cut the DNA and the other added dash group. What is that group? So first of all, you need to understand how things work over here. Suppose this is a E. coli, and now E. coli will be having its own genetic material. Agreed? It will be having its own genetic material. Now the virus comes and tries to attack the E. coli. That is a bacteriophage will come and try to attack. And while it attacks, you know what happens? It sends its genetic material inside. It puts its genetic material inside. Why? Basically, it wants to incorporate into the genetic material of the E. coli. Because whenever E. coli will undergo replication, that time this thing also will undergo replication. Whenever E. coli will be producing proteins, this particular gene segment will also be producing protein. So that was the use. But E. coli found it, and E. coli said, you cannot do that with me. I will be suing you. So how did it sue? Basically, it produced two enzymes. One enzyme which was there, you call it restriction enzymes. Restriction enzyme. What does it do? It basically cuts the DNA. Restriction enzymes are responsible for cutting the DNA. They are molecular scissors. Now, ma'am, E. coli is also having its own genetic material. This genetic material can also be cut with the restriction enzyme being produced, right? So that is a problem. So E. coli thought, that does not work with me. I will basically protect myself by adding methyl group, such that whenever methyl group is added, the restriction enzyme won't be able to act on it. So that was the thing. So first was the restriction enzyme used for damaging the genetic material of other organism, and to protect itself from the attack of its own restriction enzyme, it put methyl group. One enzyme was adding methyl group. Fine. So over here, that is a question. One of them cut the DNA and other added what group? It was methyl group. Will you forget it? I hope you don't. Next question. So I've explained all of it. You can, if at all you want, you can read it. Now, restriction site of EcoR1 enzyme is, what is the restriction site? Do you know? Restriction site is the site where the EcoR1, eco comes from E. coli, okay? E is basically the first letter of the genus name. CO is the first two letters of the species name. R is basically from which strain are you getting it, okay? R basically shows from which strain are you getting it from, and 1 basically is making us understand the time of discovery out. Like, so the first enzyme that has to be discovered, the second enzyme that is discovered, and things like that, okay? So EcoR1 enzyme site is what? It is 5-GAA-TTC3. So 5-GAA-TTC3, this is the thing, GA-TC. Okay, now over here it actually makes a cut between G and A. It actually makes the cut between G and A. That is a site, but in that specifically making a cut between G and A. Do not forget it. Now, what was the first restriction enzyme to be isolated? Do you know the first restriction enzyme which was there? If at all you do not know, please remember it is HindII. HindII also has a restriction site of 6 base pairs. So here also we have 6 base pairs. Similarly, HindII also has a restriction site of 6 base pairs. Chalo. Which of the following is the correct representation of primer annealing step in PCR? Primer annealing step in PCR. What does PCR do? PCR helps me in replication of the little amount of DNA that I have. Okay, it helps in replication of the little amount of DNA that I have. So over here, my dear children, when we have the actual segment of DNA, suppose these two strands are there. First of all, my job will be to separate them. Okay, and after separating, I want to make a copy of this DNA, right? And DNA polymerase cannot start off by itself. It always needs a primer. It always needs a primer. On this primer, the DNA polymerase would be starting working on and giving rise to a whole new DNA strand out there. Now the thing is, you should know DNA polymerase works from which direction to which direction. Always remember, it will be working from 5' to 3' direction. So in that case, on the other side, what should be the direction of the DNA? It should be 3' to 5'. So primer is going to come and attach on the 3' end. Okay, and the DNA polymerase will be working from here to here. So from 5' to 3' direction, it will be making the copy. Similarly, my dear children, suppose this is the 3' end, this is the 5' end. The primer will be coming and dashing over here and making a new strand in 5' to opposite to this, 5' to 3' direction. So which of the following is the correct representation of primer annealing step in PCR? So you should know the primer should be coming and attaching on the 3' direction, and it will be on the opposite sides. It cannot be on the same side because these two strands, one run from 3 to 5, another run from 5 to 3. So it will be opposite places. So over here, what will be your answer for this question? Here it is a 3', here it is a 5', so this is wrong. Here it is a 3', but here again it is a 5', wrong. Here again 5', wrong. Here at 3' position, Here also a 3' position, which is why your answer becomes this. And this process of basically putting down or combining the primer out there, you call this as annealing. You call this as annealing. So first step is denaturation, where you separate. Second is annealing, where you attach the primers. Then there is extension, where basically you are extending the DNA segment with the help of DNA polymerase. What is the name of the DNA polymerase used in PCR? It is known as Taq polymerase. Why Taq polymerase? Because you get it from a bacteria which stays at high temperature. So T stands for thermus, AQ stands for aquaticus. So thermus aquaticus DNA polymerase is being used over here because this whole process is happening at very high temperature, 95 degrees Celsius, 70 degrees Celsius, things like that. So you should know that. Okay. So this is the steps that are involved. Please go through the picture very, very well. Denaturation, annealing, extension. And with that, I remember one thing, that is, what was it? So you have got a little pathogen. Now, okay, which is that particular process in which you can even identify a small amount of pathogen? With the help of what technique can you identify the small amount of pathogen as well? It is PCR, because with PCR you can amplify it, you can increase its number. Always remember that. So which of the following is the correct order of processing of PCR, my dear children? Tell me now. Annealing? Wrong, ma'am. Denaturation first, ma'am. Then extension? No, ma'am. First you have to put primers. So denaturation, annealing, and then extension. Your answer for this question would be C. Cool? Cool, ma'am. Chalo. In the given diagram of PBR 322 vector, identify the restriction sites A, B, C, D. A, B, C, D. So this picture is super important. You would be asked to draw it. Make sure you know it. I'll tell you how to do. You call this as ORI site. This is where the origin of replication is. ORI stands for origin of replication. This is where the DNA polymerase will start replicating, making the new DNA strand. Now for that, it needs a protein. For replication, it needs a protein, and that protein site, that formation of protein site is known as ROP. What do you call? ROP site. Now I should not be putting square around it. This is the ROP site. This is the ORI site. Now my dear children, you know what? This particular PBR 322, it has a gene. It has a gene. If it codes, okay, if this gene is able to express itself, what will happen? This will provide antibiotic resistance. Which antibiotic resistance, ma'am? It will provide ampicillin resistance. So ampicillin is basically an antibiotic, and this antibiotic, my dear children, suppose I grow a E. coli that has PBR 322 having this particular gene segment, it will be resistant to ampicillin. So even if I put ampicillin, it will be easily able to grow because it is resistant to ampicillin. Now on the other hand, you have another gene which will be coding for resistant to next antibiotic, which is tetracycline. So I'll write it tetracycline resistant gene. Okay, tetracycline resistant gene. Now, I am pretty sure you might have all heard about some enzymes which come and cut things, restriction enzymes, right? Why do you need to cut it? Basically, we make a cut and we insert our own gene of interest out there. That is a thing. That is a use of, what do you call this? The restriction enzymes out there. Now, there are some sites over here for restriction enzyme. I'll start from here. So, always remember, here you have 1. On both these sides you have 2, and at the top you have 3. So 1, 2 and 3. Here you have the site for a restriction enzyme whose name is PVU1. PVU2. PVU2. This is the restriction enzyme site. Now where is it coming? In the site of ROP. Now suppose I am using restriction enzyme PVU1 for cutting, and it cuts, okay, it makes a cut out there. Do you think it will be still able to produce this ROP protein out there? No, it won't. So the function is lost. The function will be lost. Similarly, I told there are 2 over here. What are these 2? It is BAMH1 and SAL1. BAMH1 and SAL1, my dear children. Not 1 as in 1, it is Roman letter 1. These 2 enzymes are up over here. Now suppose I cut with the help of BAMH1. So that means it is making a cut over here. Now do you think this plasmid would be resistant to tetracycline anymore? No, it won't be. If I put tetracycline media, this particular organism that has this plasmid would be killed because it is not resistant to tetracycline anymore. What do you call this process? when you insert something, it is getting deactivated. You call this process as insertional inactivation. Insertional inactivation. You are activating something by inserting a gene of interest, and that happens when you have restriction enzymes out there, restriction enzyme sites out there. Now similarly, I told you 2 of them are here also. What is this? So we heard about PVU2, right? Here you have PVU1 and also you have PST1. PVU1 and PST1 site is over here. Again, if I use PVU1 for cutting, then my ampicillin resistance would be gone. Cool. I told you there are 3 genes over here, 3 restriction sites over here. One is for the very famous EcoR1. There are 2 of them which looks like this. You call it as CLA, okay. You talk it as CLA2 and HIND3. So CLA2 and HIND3. So CLA2 and HIND3. Please remember that. Cool. Just give me a second. Sorry, CLAV1, okay. CLAV1, not 2, CLAV1. I'm so sorry. That's what I was a little bit confused. So it is CLAV1 and HIND3. Okay. 3, 1, 2. 1, 2, 3. Fine. Do not forget the number just like how I did. So it happens. So 2 is over here, 1, both of them are 1, 1, 1 only. Here also 1, 1, 1. Here you have 3. Here you have 3. So the entire structure, it looks like this. Over here, you have been asked about A, B, C. So A is origin of replication, ORI site. Okay. What is B? Ampicillin resistant gene, or probably, yeah, ampicillin resistant is given over here. What is C? Tetracycline resistant gene. What is D? D is one side, right? So it will be HIND3. So this is the actual picture. You can follow it. Okay. Okay, ma'am. Chalo. Which of the following is not a feature of plasmid? Which of the following is not a feature of plasmid? Plasmid is what, my dear children? Suppose E. coli is there. E. coli will be having its own genetic material. Now it has some extra chromosomal material out there. What is it? It is some extra chromosomal material. That means my genes which are required for formation of a proteins that help me sustain are not found over here. This extra chromosome. Whatever I need for sustenance, that genes have been coded by my actual DNA or by my active genetic material. This is the extra chromosomal content which is there. It is usually circular in shape. It is never single stranded. It is double stranded, my dear children. So this is wrong. Independent replication, does it show? Yes, because it has its own origin of replication. So replication will have independently. You do not have to rely on anything else, but it is not single stranded. So this is not the feature of a plasmid. Cool. Done, ma'am. So this is what I have given. They might have selectable markers, they might have antibiotic resistant genes, all of it. Fine. Agrobacterium tumefacient transforms normal cells into a tumor cell by delivering a piece of DNA known as. So Agrobacterium tumefacient, which is there, is a bacteria. Now this particular bacteria is responsible for transferring transforming normal cells to tumor cells. Okay, transforming normal cells to tumor cells. But how do they do that? So this particular bacteria, my dear children, it also has a plasmid. And do you know the name of this plasmid? This is known as TI plasmid. The name of this plasmid is TI plasmid. TI stands for tumor inducing plasmid. Okay. So how does it induce tumor, ma'am? So there is a gene over here which you call as tDNA. You call this as tDNA. T stands for transfer. T stands for transfer. So this particular plasmid has a transfer DNA in it. Now what usually happens is that when this bacteria affects a plant, it transfers the tDNA into the plant. And this tDNA, my dear children, is responsible for the formation of, or for the cause of tumor in plants. So tumor inducing plasmid is there. In that, what is the inducing substance? That is the tDNA or the transfer DNA. Okay. Now suppose, how do people use it for their own good? Why not just remove the tDNA? Why not just remove the tDNA and instead of that put my gene of interest? Put my gene of interest. Anyway, the bacteria is going to affect the plants, but while it affects the plant, it is going to pass down my gene of interest out there. So which is why we say Agrobacterium tumefacient is the vector for plants. Always remember, what is a vector for plants? It is Agrobacterium tumefacient. Okay, so here by delivering what piece of DNA? It will be transferred DNA, or what you call as the tDNA. Now, the purpose of stirred tank bioreactor is, what is the purpose of stirred tank bioreactor? Have you seen the stirred tank bioreactor? It is there for mixing out everything. By mixing out everything, I mean the mixing out the nutrient content that we are giving. Apart from that, mixing the oxygen. If at all oxygen is not there, do you think the bacteria will be able to grow out there? No, it won't be. So that is the thing. So providing anaerobic conditions in the culture vessel, wrong. Purification of the product, no. Addition and proper mixing of the preservative, somewhat yes. But availability of oxygen throughout the process, that is super important. Otherwise, the bacteria are not able to grow. So stirred tank bioreactors purpose is this. Please don't get confused between this and this. The answer for this question is D. This is a stirred tank bioreactor. You might be asked to draw it as well. Make sure you go through the picture really well. Make sure you know the labelings. So over here you have the, what do you call, flat blade impeller for mixing things. Here you have the sterile air which is being put, and this impeller, when it moves, it gives it to all the sides. There is steam for sterilization of everything. Whenever the things are being removed, you need to sterilize it. There is a place for pH control. This is the motor which runs the impeller. You have something like this. This is important. This is known as foam breaker. It does not allow the foam to form over here. The foam is not formed over here because of the foam breaker. Okay, and the culture medium is there. That's all. Timer, everything is there. Cool. Next question. Identify the correct statement. Hint 2, cut the DNA to specific sequence of 6 base pairs. I told you hint 2 is the first, restriction enzyme to be found out. And yes, it has a sequence of 6 base pairs. This is correct. Micro-injection is a technique of introduction of our DNA into the plant cells. So my dear children, when we want to put recombinant DNA, what do we use? We use usually vectors such as agrobacterium tumefaciens. I already told you. But now, if at all you want to insert specific type of DNA into the plant without a vectorless method, if at all you're talking about, how can you inject it? So for that you have another technique which is known as biolistics. So here the options are wrong. Understand, biolistic which is there is a technique which you also call as gene gun method. Gene gun method, they come under vectorless transfer. Okay, vectorless methods of transfer. So why do you think that we need a gun to transfer things into a plant cell? Because plant cell, my dear children, have an additional wall layer which is known as cell wall. And to permeate into that, you need a gun. So basically, the gun is being shooted and there are balls of gold and tungsten. Okay, gold and tungsten. Filaments would be there. Oh my God. Gold and tungsten would be there, my dear children. Okay, and wrapping around it, you have the DNA. It is being shooted and it directly goes inside the nucleus of the plant. This is known as biolistic or gene gun method. Whenever you do not need a vector like agrobacterium tumefaciens, you can do this. Not you, basically the people who do this, do this. Now there is one more method for animals. For animals, my dear children, do they have cell wall? No. So it is pretty easy for letting things inside. For that, you have a technique which is known as micro-injection. Okay, you have a technique which is known as micro-injection, which basically transfer the genetic material inside the animal cell. That is for micro-injection. This is a vectorless method. Ma'am, do we have vectors for transferring it? Yes. So just like how you have agrobacterium tumefaciens for plant, in animals, what is the vector which usually infects us? Viruses. Viruses know to transfer their genetic material, right? So why not put whatever we need inside the virus and then ask it to transfer? It will do, right? Not ask it to transfer as in, hey virus, can you transfer it? No, it's just using its own natural mechanism to transfer it. Okay, so basically the vector using method is retrovirus. We can use disarmed, okay, not disarmed means they are not going to cause any trouble to us. So disarmed retroviruses can be actually used as a vector. Now if at all you want a vectorless method, then there is micro-injection. So micro-injection is a technique of introduction of RDNA into the plant cell. No ma'am, it's animal cell, what are you telling? So this is wrong. Biolistic is a technique of introduction of RDNA into the animal cell. Ma'am, it is plant cell, no? So this is also wrong. Biolistics has usually been forgotten. Please remember, it is the same name for gene gun. Now, retroviruses are commonly used cloning vectors in plants. It is not plants, it will be us, animals, humans, everyone, okay? Not plants. So, correct statement is only A and that's called. Okay. Next. So, here is the whole answer. If at all you want, you can go through it. Okay, now we are going to the long answers that come from biotechnology principles and processes. It might take a bit of a time, but it is definitely going to be useful. Chalo. Explain the process of preparation of competent host for transformation with recombinant DNA. Competent host as in host which is capable of taking the new DNA. So when we say that we want to make a host competent, that means we want to make it capable of taking in whatever we have for it. Okay, so we have to tell the process of preparation of a competent host. How do you make a host competent to take up the DNA out there? Do you know the process? I'll tell you. So for that, first of all, you need to have some calcium ions, just like how do you make food, right? Recipe. Just like that, we need to have some divalent cations, that is basically calcium ions. Divalent as in two valency, divalent cations. So Ca2 plus is our thing. Now, why do you think that we are going to treat, okay, we are going to treat a host with Ca2 plus? Because we want to neutralize the negative charges on the DNA as well as the cell membrane. Not the DNA, basically cell membrane. So for example, I have a cell, I want to make sure that cell takes up the genetic material. I will make sure it is being treated with calcium Ca2 plus. Why? Because I want to neutralize the cell membrane. Our cell membrane is not neutral. We want to neutralize it. Otherwise, what will happen, my dear children? The DNA which is negatively charged, it won't enter. And not just that, we have to neutralize the cell membrane and also the DNA which is actually negatively charged, so that things can be easily taken up. So Ca2 plus ion neutralizes negative charges on the DNA and the cell membrane. So suppose I have a cell over here, I have the DNA which I want to insert over here. Both of them are being treated with calcium 2 plus. Okay, Ca2 plus. Now the mixture of cells and the DNA is then given a heat shock. So first of all, what do? I put it in a cold medium. Okay, and then I'm such a psycho, I'm going to change it and put it into heat shock treatment. I will be giving it heat shock treatment. That means I will increase the temperature to 42 degrees Celsius for 90 seconds. Why do you think this is being done? Poor cell out there, right? Why do you think this has been done? So that there are some transient pores being made, some temporary pores being made. Why do you think we need temporary pores? Because we want the DNA to get entry into it. Now, once it has entered with the help of temporary pores, what will happen? I will put it again into cold. I will put it back into cold thing. Why? Because I want to close the pores. Okay, so preparation of competent host for transformation, this is usually done with bacteria, my dear children. Bacterial cells are treated with divalent cations to make them competent. The Ca2 plus ions neutralize the negative charge on DNA as well as the cell membrane. The mixture of DNA and the cells and the DNA is then given a heat shock. Before that, you need to cool it on ice. Then you give heat shock. Then again you cool it on ice. This creates temporary pores, allowing recombinant DNA, the DNA that we want, into the bacterial cell. Got it? Yes, ma'am. Got it. Next question. Mention the restriction enzyme that can be used for insertional inactivation of antibiotic resistant genes. So over here, they are asking you, can you tell me the name of restriction enzymes that can be used for insertionally inactivating the antibiotic resistant genes? So over here we have two resistant genes and we want to inactivate it. So what enzymes will you use? For inactivating tetracycline, you can use BAMH1 and SAL1 because both of them have the restriction site over here. For inactivating ampicillin, you can use PVU1 or PST1. Got it? So that can be done. So that is the answer. BAMH1, SAL1 for inactivating tetracycline resistant gene, PST1 or PVU1, not PVU2. Oh my god. Okay. PVU1 inactivate the ampicillin resistant gene. Now one more question is there. How many cloning sites are present in the given PBR322? So how many cloning sites do you think are present in this particular PBR322? Cloning sites means restriction sites where I can put my DNA. I have 1, I have 2, I have 3, 4, 5, 6, 7, 8. How many? I have 8 of them. I have 8 of them. Okay, and with that I remember 1, 1, 1, 1. All of them are 1. This is 3. Again 1, 1, 2. So 1, 1, 2. Again 1, 1, 3. If at all you want, you can use that. I just came into my mind. Anyway, there are 8 cloning sites out there. Now give reasons for the following things. Okay, restriction enzymes are called molecular scissors. Why do you think restriction enzymes are known as molecular scissors? Because they are responsible for cutting They are responsible for cutting the big molecules out there, such as DNA, RNA, stuff like that. So because they recognize and cut a specific base sequence, please give an example of EcoR1. The EcoR1 cuts between in the restriction site 5-GAATTC3-, cuts between G and A. Now DNA ligase is used in genetic engineering. DNA ligase is used in genetic engineering. Why DNA ligase is being used? So my dear children, when we cut things and when we put the new thing, obviously there might be some space out there. You need some fevistick, right? And that fevistick's role is being played by DNA ligase. It is used for ligation of the DNA. So because it joins two DNA fragments by forming phosphodiester bonds. Between the nucleotides of the DNA, what bonds do you have? You have phosphodiester bonds. So you help in, this particular enzyme helps in the formation of phosphodiester bonds between the sugar and the phosphate backbones, helping to seal the foreign DNA to the new vector, to the new place where it has been inserted to. Now, chilled ethanol is used in DNA isolation. So suppose, my dear children, you treated the cell with RNAs, proteinase, lot of things, and now you have DNA in there along with some things. So how do you separate the DNA from everything? You put something that is known as chilled ethanol. Chilled ethanol is put such that the separation of DNA can be easily happen. They settle down. The DNA is going to settle down when these things happen. So because DNA is insoluble in cold ethanol, not settle down, it precipitates out. It precipitates out from the cell extract, making it visible and easy to collect. So here I have a picture. This is chilled ethanol. Look, what is happening? Isolation of DNA is being happening. So it is not sedimenting down, basically it floats around. Okay, but anyway, it is precipitating, right? Now, smaller DNA fragments move farther in a gel slab. Why do you think smaller DNA fragments are going to move faster? Because they can easily move through. They are small substances. They are smaller substances. They will be easily moving. Over here, always remember, here you have the positive thing, that is the cathode. And here you have the negative region, that is known as anode. What is our DNA? Our DNA is negatively charged. Sorry. Here we have the negative, oh my god. Cathode is there, but this is negatively charged, and here we have anode, which is positively charged. Our DNA is negative, which is why it will move towards the positive side. It will move away from the negative and go towards the positive side. Always remember, anion is negative, but anode is positive. Okay, so it will move towards the anode. Now over here, because during gel electrophoresis, smaller fragments face less resistance. Yeah, that is also a thing. It faces less resistance. If at all you're talking about bigger fragments, obviously they will face more resistance. And therefore migrate faster and farther towards the positive electrode. One more thing. In gel electrophoresis chamber, they do not move on the surface. Always remember, they do not move on the surface. Rather, the materials which are going to move, they are going to show a sieving effect. Sieving effect as in within the material, within the gel it is going to move. Within the gel it is going to move. Now ma'am, I want to see the DNA fragments which are being separated. What is the thing that I can use for viewing it? What do you use? You use ethidium bromide. You generally use ethidium bromide for visualization. Under UV light, ethidium bromide comes to give a color like orangish reddish color would be given. Okay, brickish orange or something like that color is given over here. Always remember ethidium bromide. Don't get confused with chilled ethanol. Chilled ethanol is basically for separating the DNA. Now, thermostable DNA polymerase is used in polymerase chain reaction because all the reactions that are taking place in PCR, it is happening at high temperature, 95 degrees Celsius, 75 degrees Celsius, things like that. That is why you need a thermostable, something that will be stable at high temperature. So these, because enzymes like Taq polymerase can withstand the high temperature, used to denature the DNA strands, allowing the PCR to continue without the enzyme breakdown. Otherwise, the enzyme would be disoriented. Cool? So this is gel electrophoresis. You can see that they are separating them, and over here the work of ligase, the gluer. See, it is coming and ligating. It makes the phosphodiester bond. Cool? Chalo. 45th question. Genetic engineering is a core technique of biotechnology. In the background of this, complete the following table. So over here you have gel electrophoresis. Gel electrophoresis A helps in visualization of DNA fragments. What is that? I told you it is ethidium bromide that you put for visualization of DNA. Now dash DNA polymerase dash DNA polymerase dash. So what is the name of the technique? PCR, my dear children. That DNA polymerase is there for what? For extension of the DNA fragments out there. Okay. Now here gene gun method is there. Biolistic. Gene gun method, what happens over here? Transfer of recombinant DNA. What do you use? You use gold and tungsten filaments. You use gold and tungsten filaments. So it is being asked what is being used over here. And there, what is the function of it? Isolation of DNA. For that, what do we need? We need chilled ethanol. It helps in precipitation of DNA. So all of it looks like this. Gel electrophoresis, ethidium bromide helps in visualization of DNA fragments. PCR, DNA polymerase again helps in amplification of the DNA. Gene gun, you use tungsten or gold particles coated with DNA, helps in transfer of recombinant DNA. Isolation of DNA, chilled ethanol, precipitation of DNA. So you just have to mention A, B, C, D and tell your answer. Okay? This is in this year's model question paper. Now, okay. Briefly explain the steps of recombinant DNA technology. Now, you might be asked to make the diagram of it, or probably you might be asked to write the whole thing. Please know how to do it. I'll just give you a brief intro about it. So, isolation of DNA. How do you isolate the DNA? Suppose, my dear children, you want to isolate it from a fungus. Fungus have a cell wall. What is cell wall made up of? It is made up of chitin, which is why we will need an enzyme that is chitinase. We will need an enzyme that is chitinase. Suppose the next thing that we have, it is a bacteria. Bacteria cell wall is made up of peptidoglycan. Peptidoglycan. For that, you need an enzyme that does not have resemblance with the name, but it is known as lysozyme. Now you have what? You have plant. Plant. Plant also has a cell wall made up of what? Cellulose. So what do you need? You need an enzyme that is cellulase. You need an enzyme that is cellulase. Now you get inside. Inside you find RNA, protein, everything. For degrading the RNA, you have RNase. For breaking down the protein, you have protease. Okay. And then finally, you separate the DNA from all of these things with the help of chilled ethanol. So DNA is isolated from both donor and the vector organism. Enzymes like lysozyme for bacteria, cellulase for plants and chitinase for fungi is being used depending on whether the cells are bacterial, plant or fungal. And basically you use chilled ethanol for separating it out. Now cutting of DNA using restriction enzyme. So once DNA is got, you cut the DNA with the help of restriction enzymes. The restriction endonucleases, please remember there can be a question coming asking the difference between restriction endo and exo. Endonucleases cut the DNA within, okay, cuts from the within. Exonucleases, they cut at the end. They usually cut at the end, always remember that. So restriction endonucleases cut the donor and the vector DNA at specific recognition sites, producing sticky ends. They produce sticky ends. So over here I am cutting the DNA of the fungi with the same restriction enzyme. I am going to cut the DNA of the vector. So I am going to get an end which is known as sticky end. These sticky ends basically they do not have. Sticky end basically means that I have base pairs over here. I do not have any complementary base pairs. Suppose other vectors DNA which is there, it is the complementary part to it. Both of them will be binding. So that is why we use the same restriction endonucleases such that we get sticky ends. So that can be a question. How do you get sticky ends? Please go through it. Now separation of DNA fragments, gel electrophoresis. After that, after you cut the DNA, my dear children, you have to make sure the smallest one is being taken which is of use for you. So the cut DNA fragments are separated by agarose gel electrophoresis based on size and the charge. These desired DNA fragments is this extracted from the cell, from the gel. So basically you get it, you use ethidium bromide to visualize it and later you want to extract it. How do you do it? What is the name of that process? That process's name is elution. This is a very important thing. Elution. It is elution, not ILLUSION. It is elution, ELUTION. Okay, that's how you take it out. Now, once you have taken it out, my dear children, insertion of desired DNA into the vector. So, you do not do it directly. Here, you will do PCR also. You will amplify. You will also amplify the DNA that you have got because it cannot be success every time, right? So then after you have amplified it, you are now going to put it in the vector. So the purified DNA fragments is joined to the plasmid vector using DNA ligase forming the recombinant DNA. Now what do you do? Introduction of recombinant DNA into the host. So now we have put it in the vector. Vector is different, host is different. Vector is someone who is going to carry it. Host is someone where you are going to put the whole thing out there. Okay, so introduction of recombinant DNA into the host cell. So recombinant DNA is introduced into the host organisms by methods such as transformation, microinjection, electroporation or bioelasticity. So transformation, microinjection, electroporation, bioelasticity, whatever you can remember, please put it out there. Now, selection of recombinants. Now, not all of them, my dear children, would be having the recombinant DNA. We think that they will not be having, but they might not be having. So, cells that have successfully taken up recombinant DNA are identified using selectable markers. For example, antibiotic resistant gene. I know that basically I have inserted my gene in the antibiotic region, so it won't be resistant. Now, if it is showing resistance, that means it is not a recombinant. So, they are selectable markers out there. They will help me select things. Okay. Now, after that, please here blue, white, green screening is also there. That is about lag Z. Please go through it once. Okay, please go through it once. I think if I explain that also, it might take too much of your time. But if at all you have trouble, tell me in the comment section. Okay. Now, expression of the gene. The host cell machinery expresses the inserted gene, leading to the production of the desired proteins. Now, expression is happening, desired protein is being produced. Now, what do you do at the end? You do downstream processing. What do you mean by downstream processing? Basically, it includes separation as well as purification. Separation of all the content and also the purification is known as downstream processing. Apart from that, later on, you are going to add preservatives and everything and do quality testing, things like that. So, separation and purification comes under downstream processing. And my dear children, you're going to use preservatives and things like that later on for keeping it preserved. Now, over here, what you said? Yes, selection of the recombinants, and then one more point is there, you have to grow them in the culture medium. You have to grow them in the culture medium. So, please put that as well over here. In the straight tank bioreactor, in sparse tank bioreactor, all of it. Okay. Now, my dear children, yes, I think that's up. So, that's all for the chapter Biotechnology Principles and Processes. Yes, now we will be moving ahead with the next chapter, that is Biotechnology and its applications. I know this video is long, but I'm pretty sure it is helpful for you. If at all you think the same, let me know. Okay. So, given below are the steps in the gene therapy of a patient with adenosine deaminase deficiency. So, you know what is gene therapy? Suppose an organism who is there who they do not have a particular gene. So, obviously, if this gene is not there, the protein which this gene used to make will also be not there. So, that is a very big trouble. So, what do we do, my dear children? We have some technique for it that is known as gene therapy, and I'll tell you how does it work. So, in a 4-year-old child, a deficiency was found, the deficiency of the enzyme ADA, adenosine deaminase. This particular enzyme is super important. Okay, this particular enzyme is super important for making sure that the cell is not in a toxic environment. It breaks down the toxic substances in the cell. And it is mostly important in the case of lymphocytes, that is our immune cells out there. So, all of the cells which is there, they have ADA, and in them the function is the same only for making sure that it is not toxic inside. But in the case of lymphocytes, it becomes all the more important. This ADA's role becomes all the more important. Now, suppose in my cell, in my lymphocytes, there is no gene because there is some mutation that have taken place, deletion mutation have taken place, this gene is not there. Obviously, I would not be able to survive much longer. Why? Because my immune system is not going to work that great. So, what can I do? Why not take the lymphocytes from my body? First of all, take the lymphocytes from my body. So, first step is taking the lymphocytes from the body. Now, after the lymphocytes are being taken up, I have to make sure that I culture them. I culture them as in I want to increase their number. So, lymphocytes are taken, they are being cultured in a culture medium, and after that what do we do? With the help of a vector. What is a vector for human cells or animal cells? Retrovirus, disarmed retrovirus. So, with the help of a disarmed retrovirus, I am going to inject the gene within my body. I am going to insert, not inject, insert the gene within my body, because this virus is going to affect in a way that the gene is being transferred. Now, I have ADA gene in my lymphocytes. I am super happy. What do? I put it back inside my body. And I have my lymphocytes which have the gene which makes it all safe. But you need to understand one very important thing. These lymphocytes have a dedicated time lifespan. They will die after some time, which is why you need repeated transfusions. Okay, which is why you need repeated transfusions. So, this process is known as gene therapy, and that is the best part of recombinant DNA technology. We have evolved in a way that we can save people. Okay, so given below are the steps in gene therapy of a patient with adenosine deaminase deficiency. Which of the following show the correct order that those steps must occur? So lymphocytes are grown in a culture outside the human body. This is done later. Lymphocytes are altered with the functional ADA gene. This is also done later. Lymphocytes are collected from the blood of the patients. Yes, this is the first thing. Lymphocytes are collected from the blood of the patient. So I have two options now which starts with 3. So I'll cut out these other options. The genetically modified lymphocytes are infused back into the patient. This is the last step. This is the last step. So one that ends with 4. This is going to be my answer. Let's see. So let us see. So after 3 is 1. Lymphocytes are grown in culture outside the human body after being taken up. Yes, ma'am, correct. Then lymphocytes are altered with the functional ADA gene. Yes, it is. Then, my dear children, what happens? Genetically modified lymphocytes are infused back into the patients. So what becomes my correct answer? C becomes my correct answer for this particular question. Got it? Cool? Yes. And also please do not forget that the type of gene that you are inserting is cDNA. Do you know what is cDNA? Complementary DNA. That means usually what happens, we have DNA, the DNA transcribes into RNA, and RNA forms the protein. In the DNA, we have a lot of things which is wanted, which is unwanted. Exons, introns are the unwanted things. But in the mRNA which is being formed, you just have the thing which will actually code for a protein. So if at all I take the DNA from someone else and put it, I will also have the wanted and unwanted regions. I don't want all that funda. So what do? I have the mRNA. This process of formation of DNA to RNA is known as transcription. Why not reverse it? From this mRNA, why not I make a new DNA, which will only have the code for making the protein? This new DNA which is being formed is known as cDNA, complementary DNA. This is what you're going to insert into the patient. This is just an extra info, but it can be asked what type of DNA is being put, so you should know. Next question. The plants developed by the fusion of protoplast of 2 different plants are. What do you mean by protoplast? So whenever we have cell wall, cell membrane, everything together, we call it as a normal cell. But suppose we are removing the cell wall of the plant. Now only cell membrane is there. You will call this as a protoplast. Now suppose you are fusing 2 protoplasts together. What do you call this process as? You call this process as protoplasmic fusion, and at the end what do you get? You get somatic hybrids. This cell belongs to a different plant, this cell belongs to a different plant. That's how you get pomato, which is a fusion of potato and tomato. So pomato, they are somatic hybrids. So plants developed by the fusion of protoplast of 2 different plants are known as somatic hybrids. Do not forget it. Ma'am, what is soma clones then? So basically, I'll tell you one very important thing. So this is a protoplastic fusion, right? Now let's forget about protoplastic fusion, or let's keep it over here. Over here, suppose I have a plant. Now this tip region, which is known as shoot apical meristem, I take this tip region, okay, and I put it in a medium where it is going to divide. Okay, ma'am, it is known as a callus now. What do you call this as? You call this as a callus now. Now you put this callus inside a medium that has rooting hormone. So what will happen? They will be forming the roots. Now what do? I put it in a shooting hormone. So now this particular thing will be having a shoot. And then you have the root and the shoot, everything. Now it's a seedling. You put it in different things. So what are these plants that you're going to get from these cells? You call them as soma clones. Why soma clones? Because all of them are going to look the same. All of them have the same genetic material because it have been taken from the same type of cells out there, which is why you've called the number of different plants arising from it as soma clones. What do you call this process as? Where you are cultivating thousands of the same type of plants. You call this process as micropropagation. This is coming under tissue culture. So micropropagation comes under tissue culture, where we take a tissue, we let it divide and form a callus, then the rooting hormone, then shooting hormone. Whatever we get at the end is known as soma clones. Now ma'am, I want to produce virus-free plants. Listen to me carefully. This is an important question. I want to produce virus-free plants. What do I do? Which part do I take? Always remember apical meristem. Okay. shoot and the root apical meristem are the ones which is going to be virus free. So you take apical meristem. Do not forget it. These cells are not going to be affected by viruses. So soma clones is different, somatic hybrids is different. Cool? Chalo. So I've explained everything out here. Okay. Identify the incorrect statement. The transgenic cow Rosie produced milk enriched with alpha-lactalbumin. Transgenic cow. Transgenic means basically you're making some changes in some organism. You call them transgenic organism now. So you had a cow whose name was Rosie. Sweet name, right? So Rosie started producing the milk with enriched alpha-lactalbumin. Is it correct or false? It is correct. It is true. You know what? Alpha-lactalbumin is the human protein. And we are making a cow produce it by doing some changes in its genetic material. So you get it? So it is an example of a transgenic cow. So it is correct. Alpha-lactalbumin is a correct example. Now, Bt toxin gene Cry1AC controls the corn borers. Ma'am, I do not know about this now, so I'll keep it on hold. Let's go. Human protein alpha-1 antitrypsin is used to treat emphysema. Is it used to treat emphysema? What is human protein? Human protein's name is alpha-1 antitrypsin. Ma'am, is it used to treat emphysema? Yes. How do you produce it? With the help of some other organisms. So again, the same gene I put in another organism and the other organism produces this protein for me and I can use it for the treatment of emphysema, which is a respiratory disorder. Okay, so both of them, they are examples of transgenic products. Alpha-1 lactalbumin and alpha-lactalbumin and alpha-1 antitrypsin. Please remember that. Now, the commercial production of human insulin was made possible by transgenic E. coli. Is it? E. coli was helping in the production of human insulin? Yes, it was. It was helping in the production of human insulin. I think we have questions related to that. Do we have? Yes, we have. So I would not be discussing that right over here. We will be discussing that later. Okay. With that, let me just see. Okay. So, what are we left with? Bt toxin genes Cry1C controls corn borers. Is it correct or not? So what is Bt cotton gene? What is this? What is that? So you know what? There is a bacteria whose name is Bacillus thuringiensis. Bacillus thuringiensis. Now this Bacillus thuringiensis we studied in Microbes in Human Welfare, right? What does it have? It has a gene that codes for an inactive toxin. You call this toxin as prototoxin because it is inactive now. It's a toxin. It is an inactive toxin. Now suppose an insect takes up this particular bacteria. This bacteria will be now reaching the gut, the mid-gut of the insect. Now this bacteria's inactive toxin is going to get activated. Now you might wonder, how ma'am, how? Basically what happens is that in the midgut of the insect, everything is alkaline, basic in nature. And when it is alkaline, my dear children, this toxin becomes what? Active. So that is the thing. Alkaline medium was not provided, then how will it become active? Okay, now alkaline medium is there, it becomes active, it is going to lead to perforations over here in the midgut region. It is going to cause trouble in the midgut region out there. Cool? Done? Fine? Now, so what have we done? Suppose, why not take this gene and introduce inside a plant cell? The plant cell will also produce the same type of toxin. When an insect injected, the insect will be dying. So these type of plants which has the genetically modified thing, that is they have the gene for Bt, you call them as Bt cotton, Bt brinjal and things like that. Now you have different type of genes. The gene's name is Cry gene, but you have different type of Cry gene. So always remember this very important technique. Cry1AC and Cry2AB. This is for first letter starts from C, so cotton. Second letter starts with B, so bollworm. Cry1AC and Cry2AB produces toxin affecting the cotton bollworm. Whereas Cry2AC, now it was 1AC, now it is, not 2AC, sorry, I'm so sorry. Cry1AB, we here use 2AB, right? So now we are going to use 1AB. This is for C for corn and B for bulls. This is for corn borers. Ma'am, both of them are starting with C. So this is a bigger word, right? Cotton bollworm is a bigger word. That is why we need two of them, 1 and 2. Over here, it's very small. That is why we need just one. We need just one, and that one first letter is C and the last letter is B. Why didn't I put it? Listen, how did I retrieve my mistake over here? How did I correct my mistake? Because I knew that if it was cry2AC, then C and C is not ending. It should be C and B, right? So if 2 is used over here, then 1 has to be there. So that is how even I remember. When there is too much things to remember, you need to have your own tricks, and I hope this helps you. So listen to it, you do it properly and write out. I think it will be helping you as well. So Bt toxin genes cry1AC controls corn borers. It is wrong or right? It is wrong, because it is cry1AB that controls the corn borer. So this is the incorrect statement. Rest all of them are correct. Okay, here it is written, different cry genes are targeting specific groups. Lepidopterans such as tobacco budworm, armyworm, coleopterans such as beetles, dipterans such as flies and mosquitoes. Now, which of the following statement is correct with respect to synthesis of insulin? With synthesis of insulin, what is correct is being asked. So I told you E. coli is used for synthesis of insulin. How? Why? So initially we used to take insulin from pigs, pancreas and stuff like that. But some people found to have allergies, started to have allergies, which is why we shifted to our only source, that is animal source. But how can we make our body produce more amount of insulin? Why not make it produce on a bigger scale? So for that, you need to understand one very important thing. Insulin, which is there, it is a hormone. It is not an enzyme, it is a hormone. And this hormone, my dear children, is a proteinaceous hormone. It is what? A proteinaceous hormone aata hai. Insulin has A polypeptide and it has B polypeptide. And both of them are combined with C peptide. So A, B and C is also there. This is the case of prohormone. This is the case of prohormone. That means I'm talking about immature insulin. Immature insulin. You need to make it work, then you have to make it mature. What happens in our body that makes this insulin, that makes this particular insulin work? It makes sure that, my dear children, there is only A peptide and B peptide. Only A and B should be combining together, and they should not have the C peptide. It should not be having the C peptide. When C peptide is removed, what will happen? This will turn into a mature form of insulin. It is no longer a prohormone. It is an actual hormone out there. So over here, I'll show you the answer, but listen. So A and B is there, in between you had C, but now you have cut off the C, you just have A and B connected to each other with the help of sulfide bonds. With the help of what? Sulfide bonds, please remember it. So maturation of proinsulin into insulin, or maturation of prohormone to the actual hormone. Prohormone, not proenzyme, I'm telling you. So which of the following statement is correct with respect to synthesis of insulin? Synthesized as proenzyme and needs to be processed to become functional enzyme. Is it an enzyme? Wrong. Synthesized as prohormone and needs to be processed to become functional hormone. Correct. Synthesized as proenzyme. Wrong. Synthesized as prohormone and needs to be a functional proenzyme. Wrong. It is prohormone which becomes functional hormone. Now, what is the role of E. coli over here? So my dear children, what did people do is that they understood that the C peptide is being cut in our body by our own machinery. But if at all you put the gene for insulin into E. coli, it won't have machinery to cut the C peptide. It would not have machinery to cut the C peptide. So what did people do? They took the gene that codes for A, and they took the gene that codes for B separately. And they put it separately. In one coli A, in another E. coli B. So now you have got the two strands. The two strands were joined together in the laboratory condition. And this was first made by Eli Lilly Company. It was first made by Eli Lilly Company. I'm telling you again, in our body, the C peptide is being removed by our machinery. We cannot expect the same to happen in an E. coli. E. coli cannot remove it. So what do? Basically, we generate A and B separately and we fuse it together in another medium out there. Okay, not medium, basically we do it by ourselves. This was discovered by Eli Lilly company, and over here your answer would become B. Now, crystals of Bt toxin produced by some bacteria do not kill the bacteria themselves, because why does not kill its own bacteria, my dear children, where this toxin is there? Because the toxin is inactive. For it to become active, what do you need? You need an alkaline medium. So this is the reason. Statement 1. The plants genetically identical to the original plants are known as soma clones. Are they? The plants genetically identical to the original plants, are they known as soma clones? Yes, ma'am. They are clones, right? Now, soma clones can be obtained through micropropagation. Can they be? Yes, ma'am. It's a type of tissue culture. So, statement 1 and statement 2, both of them are correct. Both statement 1 and both statement 2 are correct. And I just explained you before, so it's okay. Now, pollen grains of a plant with 2n is equals to 48 are cultured by tissue culture to produce a callus. What would be the number of chromosomes in the cells of the callus? So, pollen grain, my dear children, of a plant which is there, it is diploid, it is having 48 number of chromosome. Now you have not taken the plant cells, rather you have taken the pollen grain of this plant. And pollen grain, my dear children, always contains half of the content, because it contains genetic material, so it contains the gametes, which is why it will always have half of what is there in the normal cell. So if 48 was there, then in the pollen grain it will be half of it, which means it will be 24. Now if this pollen grain is used to make a callus, that means a group of cells, then all the cells will be only having 24 chromosomes, right? So your answer for this question is 24. Cool? Now, statement 1. Bacillus thuringiensis produces insecticidal protein crystals called Bt toxin. Bacillus thuringiensis produces insecticidal protein crystals known as Bt toxin. Correct. These toxins exist as prohormones before entering into insect gut. Wrong. They exist as protoxins, or protoxins, not as a hormone. So basically statement 1 is correct, but statement 2 is false. Statement 1 is true, statement 2 is what? False. So your answer becomes A. Cool? Now, use of bioresources by multinational companies and other organizations without proper authorization and compensation payment is referred as. I am using some resources, I have not taken any permission from anywhere, and these multinational companies are using it without payment. What do you call this process as? This is known as biopiracy. You can get questions like this. You may all be also asked, what is the role of basmati in this? So you know what? American company, they started patenting basmati, saying that this is our product. But our Indian agriculture farmers, my dear children, they were producing it without compensatory payment. Can you patent it or can you use it? No. So this comes under biopiracy. Okay? Okay, ma'am. Now, which of the following statement is not correct? Most Bt toxins are insect group specific. Aren't they specific? Yes, ma'am. Now, this statement, RNAi takes place. I do not know this. We'll keep it on hold. Recombinant therapeutics do not induce unwanted immunological responses in human. Recombinant therapeutics does not induce unwanted immunological response. Yes, it does not. That is why we use it. Now, adult onset diabetes can be controlled by taking insulin at regular intervals. Yes, insulin's role is to lower down the sugar level. That means diabetes can be treated with this. So this is also correct. What is not correct? Probably this. RNAi interference takes place in all eukaryotic as well as prokaryotic organism as a method of cellular defense. So my dear children, first of all, we need to understand what is RNAi interference. So what exactly happens is that there is a aschelminthes, which you call as meloidigine incognita. Now this particular worm which is there, it is going to affect the roots of tobacco plant. It gets inside the cells and it causes damage in the roots of the tobacco plant. So when it causes damage, obviously, basically it has some genes. That genes might be giving rise to RNA. That RNA, my dear children, might be coding for some protein. Now, if I want that this protein is not to be made, should not be made, what can I do? The mRNA, which is single-stranded, If I give a copy of it over here, make it double-stranded, do you think it will be able to undergo the process of translation and make proteins? No. And that is the exact thing that you use in RNA interference or RNA interference. You're interfering between the working of RNA. How do you do it? You take the gene of this particular organism. Okay, you take the gene of this particular organism, as in the gene that will be coding for the RNA. Okay. Now what do? Basically, with the help of a vector, that means in the case of plants, what is a vector? We have studied it. It is Agrobacterium tumefaciens. We take this vector, we put this particular gene, and we help it infect the particular plant. Now, the plant, my dear children, will be having what? The gene that codes for the RNA. Now, the RNA which is being produced, you need to understand, it's not single-stranded. It is going to be a double-stranded RNA. It is going to be a double-stranded RNA. Otherwise, it will also cause trouble. No. So, double-stranded RNA is being produced over here. Now, what happens? This double-stranded RNA, when it comes in contact with the mRNA, of Meloidica incognitia, which is trying to cause trouble, this double-stranded RNA, my dear children, or I'll make it in white, this double-stranded RNA is going to separate. They are going to separate, and one of them comes over here, binds with the mRNA, making it double-stranded, and hence you have interfered it. So what do? You take some genes which is responsible for coding double-stranded RNA. With the help of Agrobacterium tumefaciens, you are going to infect the plant, you pass the gene, and it will have double-stranded RNA being formed. Now, the cells where mRNA is being produced of this particular organism, double-stranded RNA will be forming a bond out there. So one strand goes, disintegrates, the other strand which is there comes and makes a bond over here. Now, this is actually a natural defense mechanism that we have as in eukaryotes have. Prokaryotes does not have this as a natural defense mechanism. So RNA interference takes place in all eukaryotic as well as prokaryotic organism as a method of cellular defense. Wrong. Only in eukaryotes. It happens only in eukaryotes. You need to know that. Now this is a long answer. This is actually a long answer. So I'll just quickly move on over here. So I explained you a lot of things in the 5 marker. One was this. So what is RNA interference? How does RNA interference help? I told you everything. Identify the nematode gene. Nematode as in the organism that is causing the trouble, Meloidica incognitia. Using the Agrobacterium tumefaciens to insert the gene. You insert the gene with the help of Agrobacterium tumefaciens. Production of double-stranded RNA in the plant. In the plant, the double-stranded RNA is being produced and then it does RNA interference. Go through it. It is super important. You should be knowing it. Okay. Now here you have one of the application of biotechnology is to get rid of the pest-resistant plant. Explain with reference to Bt cotton, just like how I explained you about Bt cotton, you need to write the answer. That cry gene is there, they produce prototoxins, and when the insect takes it, it gets into the midgut. In the midgut, it leads to perforation of the epithelial cells, and that's how the organism dies. So please know about all these chotu things as well. We have discussed that. Now, one more question is there. Why are meristems preferred in tissue culture? I told you that they are actively dividing. Apart from that, they are usually virus-free. This is one of the most important thing. What are the things that you can use for virus-free production of plants? It is, you're going to use the meristematic cells for tissue culture. Okay, I'm jumping to this because I have explained you this already. Now, explain the applications of biotechnology in the field of medicine. You know how insulin is being produced. I told you about that. So, insulin and also the gene therapy incident, you have to talk about that. First success was in the year 1990. Okay, so I explained all of it. Whenever you have the diagram to be made, make sure you know this diagram very well and you draw it. This we have not discussed yet, so we will go back to what we were discussing. Yes, this is the last question anyways. So for MCQs. So match column 1 with column 2 and select the correct option from the given codes. Alpha-1 antitrypsin. Alpha-1 antitrypsin is used to treat emphysema, respiratory disorders. Say A goes with 3. A goes with 3. I just have 2 options. Let me cut these options out. Transposon. Let's keep it on hold. Retroviral vector. Why do you use retrovirus, my dear children? For an animal cell, for being a vector for animal cells. So gene therapy is somewhere where you need retroviral vector. Now what is ELISA? ELISA is enzyme linked immunosorbent assay, which helps me in finding out if someone has AIDS. It works on antigen-antibody principle. We discussed it, right? Antigen-antibody principle. What are we left with? Transposon. Transposons are mobile genetic elements. That means they can move from one area to other in our genes. So transposon goes with this. So what is your answer for this particular question? Your answer would be P. Okay. Now the 5 marker. Yeah. Genetically modified plants have been developed through core techniques of biotechnology. How are these genetically modified plants useful to human beings? So you have to tell. Human beings, basically farmers, they need to rely on little nutrients. If at all, they do not have to purchase big body of nutrients, my dear children. If the plants can sustain with little amount of nutrients, their mineral usage is really well. Don't you think it is helpful? Yes. So enhanced crop yield and efficient mineral usage. GM plants, that is genetically modified plants, grow faster, use nutrients better and produce more grains or fruits. This helps in having higher productivity from limited resources. Resistance to pest and disease, we already discussed about Bt cotton, RN interference in plants and all of it. Reduced use of chemical pesticides, because my dear children, they are already pest resistant. So why do you need chemical pesticides over here? Tolerance to abiotic stresses, that means they can survive in drought area, in area with higher salinity as well. Now, improved nutritional quality. Genes can be added to increase the vitamins, minerals, etc. For example, have you heard about golden rice? So golden rice is there. Why do you call it golden rice? Because, my dear children, it has vitamin A in it. Vitamin A in it. So I am basically having a rice which gives me vitamins, and this was super useful for people who were facing malnutrition in Africa. Okay, so improved nutritional quality with the help of introduction of all these vitamins, minerals and everything. Reduces post-harvest losses. After the harvest also, the storage can be difficult, it can get fungus and things like that. But since it is pest resistant and it is resistant to a lot of things, my dear children, it can slow down the process of spoilage as well. So these are the points that you need to write. Okay, now briefly explain the use of transgenic animals. This is the last question for the day. So application of transgenic animals, study of normal physiology and development. So basically, suppose I have a gene and I do not know its function properly. So I can insert the gene in an organism and see what changes are there in the organism. I'll get to understand how the gene is going to regulate, what is its effect on the natural or the normal body functions out there. So this is one thing. Second, disease study and human models. So created as models for human diseases such as cancer, cystic fibrosis, arthritis and Alzheimer's, and I'll see what are the changes that happens in the body, what are the areas that I should be looking forward for, what are the medicines that I should be giving. Okay, so when you understand what are the issues happening in a body, you will be able to find the cure better. So for disease study and human models, that is being used. So because mice, my dear children, one very important thing, what is the frequently used transgenic animal? It is mice, because it has so much similarity with our genetic system. Okay, so cancer, cystic fibrosis, arthritis, Alzheimer's, please remember their name are also important. Biological products, for example, human protein, alpha-1 antitrypsin. For treatment of emphysema, you get it from transgenic animals. Rosie, the transgenic cow which was there, gives us alpha-lactalbumin, a human milk protein. Now for safety testing of vaccine. So vaccine I put in an organism and see how does it work. Is it causing any trouble in the organism? So in that way also. Toxicity testing, that this much dose, I'll make the organism sensitive. Now then when I give doses of toxin, I'll get to understand that even in this sensitive organism, it is reacting this much. So probably I should be making it less toxic. I know it's so bad. I know it's so bad. I feel bad while saying it, but that's how it works. Okay, now there are so many techniques, other techniques being involved such that you do not use animals for all these things, but yes. So that is the use of transgenic animals. Go through the examples really well and the names of the diseases as well. Rest all of it, my dear children, we have discussed. And with that, finally, we end today's session. I just want to say one thing. Please give your best. And when I say that you have to give your best, that means please study as much as you can. Do not drain yourself. Sleep at time. And do not have the overburden of things as well. Do not get tensed. I know it's very natural to get tensed, but you can do it. Okay, remember that. Wishing you all the best. And my dear children, if at all you like this session, share it with your friends and do not forget to hit the subscribe button right now. Bye-bye.